Hello and welcome to the session number 3 of the Open Session 2020 Special Edition for the European Commission for the Control of Foot and Mouth Disease. I'm Carissa Miller, a veterinarian who's working with EuFMD, and I'll be moderating this morning's session. To introduce this session on vaccine security and critical resources for emergency management, I'd like to pass you over to Fabrizio Rosso, our Deputy Executive Secretary for EuFMD. Thank you, Fabrizio.
Thanks to you, Carissa, and good morning, good afternoon, good evening, everyone. We are today at the third session of our Open Session 2020 with the title Livelihood at Risk in a Faster World. The first week of the Open Session was focused on risk assessment and forecasting, with tools and methodologies presented for investigating the dynamics of animal mobility, understanding their driving factors, and understand how the related risk can be better monitored and forecasted. Another theme of the first week was to debate on how surveillance, control, and preventive measures can be improved in order to better address risk changes, with methods presented on how to perform evaluation of risk assessment results and how to improve surveillance and control measures with the use of predictive models, and also how to enhance communication of risk to ensure proper response. I remind you that all the recordings of the previous 2 sessions are available on the website or on the virtual tour that I really encourage you to visit. And as mentioned during the last session, it is also available at poster session, where you can find posters related to the different sessions, and also a specific session on diagnosis and virus characterizations with a very interesting research studies presented. And also a forum where you can post questions or discuss study results with the authors. Well, this week we'll be moving from assessment of risk into the capacity to respond to crisis with probably a lot of parallelism and reflections on the current COVID-19 crisis. Particularly, the session of today will address the questions related to how we can ensure sufficient critical resources, in particularly quality vaccines and diagnostics, but not only, as also the planning capacity to implement actions and the availability of related resources in response to crisis will be considered. So without any further delay, I will pass back the floor to the mic to Carissa, and I wish you all a good session. Thank you.
Thank you very much, Fabrizio. Before we continue with session 3, I just wanted to recap and give a brief overview of how this Open Session Special Edition will run. So the Open Session is a biennial event that EuFMD has been running since the 1970s, but this is the very first online Open Session. We're fortunate to be joined by more than 1,400 registered participants for this event. And to ensure that everyone has the opportunity to interact with our speakers, our partners, and EuFMD's initiatives, we'd just like to encourage you all to visit the event website, eufmdvirtual.com. There you can post questions for our speakers on the forum, which they will answer during the live roundtables at the end of each session. You can also visit a number of information desks to learn more about EuFMD and partner initiatives, including training, simulation exercises, the EuFMD disease spread modelling work, and much more. As Fabrizio mentioned, we also strongly encourage you to visit the posters. There's some fantastic work being presented in that area of the virtual tour. And you can also listen to some podcasts that summarize those posters as well. In the gaming area, you can try your hand at the Get Prepared wall or the Lesion Aging game, and all our partners have virtual areas you can visit as well. So these live sessions are streamed on Facebook and YouTube and the virtual site, and a recording can be viewed from 1700 hours or 5 p.m. CET, that's Central European Time, for those who are in different time zones or can't join us live. So let's get into session number 3. I would like to introduce to you your chair for this session, Dr. Sten Mortensen. And since 2001, Sten has been the head of veterinary research and development at the Danish Veterinary and Food Administration. The work involves the development of animal health control and surveillance strategies in Denmark for a broad spectrum of zoonotic and animal diseases. And before that, Sten worked as a researcher at the Federation of Danish Pig Producers and Slaughterhouses, where he acquired a PhD in veterinary epidemiology. Sten, over to you. Thank you.
Thank you very much, Carissa. Good morning, good afternoon, and good evening to all, and welcome to this session number 3 on vaccine security and critical resources for emergency management. Due to the coronavirus, we're all in solitude at home or where we are. Hopefully today we can feel a sense of togetherness discussing these viruses that are so important to the livelihoods of people everywhere. The EuFMD team has made a great effort to establish this virtual platform where we can come together for inspiration and discussions. Please use this opportunity to explore the content and the posters, interact, get in contact, give feedback, and get to know new people as if we were physically at the same spot of the planet. COVID-19 has been the defining virus of 2020. There are a lot of lessons to be learned from it. For my own part, we have seen how this virus could spread in the Danish mink population as fast as FMD would spread among ruminants. And we were not able to quickly enough put a stop to the virus with conventional methods before it was too late. Now nearly all the mink farms in Denmark have been called to finish the problem. Not an ideal solution. Viruses are little algorithms that do their thing when they have the opportunity. That depends on the populations of susceptible species and the conduct of people in these communities. Viruses have no legs. They spread by direct contact between susceptible species. They lure in the dirt. They float on the wind, or they're being transported by insects. What we can do is try and understand the virus. How can they be monitored and controlled by humans and societies? That also includes the understanding of why people do as they do and how. The next 2 hours in the morning session, we will start out with 2 keynotes on how to use modeling to prioritize and justify investments in getting prepared for the fast diseases, and what lessons can be learned from COVID-19 as a veterinary lab. Then we will go into depth with the early immunology of vaccination against foot and mouth disease in pigs, and see a new tool to predict the demand for vaccines in an endemic country for progressive control of foot and mouth disease. From California, we have the opportunity to get inspiration from their vaccination plan to control a potential nightmare there. And the round table with all the presenters, we will see the status. And before the round table with all the presenters, we will see the status of preparedness of the EU member states for vaccination against foot and mouth disease, lumpy skin disease, peste de petit ruminant, and sheep and goat pox. After the round table, there will be a 2-hour break before the afternoon session. Control of fast diseases will rely on vaccines, and that's what we are going to talk about in the afternoon, both in the endemic and free countries. Here we will first learn about modern vaccine platforms that can quickly provide safe and efficacious vaccines, and about vaccine banks and their role in making it possible to deliver a solution to the problem quickly. After understanding how to prioritize between vaccines to maximize impact, we will take a trip to Tunisia to hear how they address the key component of staff with skills for vaccination against foot and mouth disease. Monitoring success of vaccination is essential, and therefore we will also hear about a new ELISA test to replace the virus neutralization test for detection of neutralizing antibodies against foot and mouth disease. And finally, we will learn about intranasal vaccination against peste diphtheria. And with these words, I hope you will all enjoy this day, and I will hand over the screen and the mic to you, Carissa. Please take us into today's program. Thank you.
Thank you so much, Sten, for a really great introduction to all the presentations that we have ahead of us. First up, we have Sam Hamilton, Director of the Epidemiology and One Health section, the Australian Government Department of Agriculture, Water and the Environment. And Sam will be presenting on how much is enough, modelling and other methods to guide investments in preparedness for transboundary animal diseases. Over to you, Sam.
Thanks very much for the kind introduction, Carissa, and hello and welcome to the audience. I'm really excited to be able to speak with you today, but I'm going to ask for a little bit of patience because it's my first time ever being a YouTuber, something which my children are very proud of, but it may take me a little while to be able to interact with you all. Look, by way of introduction, first up, I'd like to acknowledge my colleague Graham Garner, who's had a very long career within our department and also spent time with EuFMD. This year marks the first, 25 years since his first publication on epidemiological modelling, and his work's really become quite groundbreaking, some of which I'll talk today, and some of the work on our latest model, ADIS, has become a backbone for EuFMD's modelling platform as well. So I'd really like to thank and acknowledge Graham as a mentor and colleague. Next slide, please. Just a quick overview of where we're coming from today. So I'll provide you with a little bit of background information about the Australian animal health context, because we are a little bit different, and all of the examples that I'm going to use today to guide investments in preparedness, particularly around resource requirements, come from the Australian context. I'll talk about a case study which involved modelling resource requirements for disease eradication within Australia. I'll talk about an older study that happened in 2007, which was a functional simulation exercise for laboratory capacity, which occurred at our National Emergency Animal Disease Laboratory, and also talk in a little bit of detail about qualitative performance assessments and standards and how we approach those problems within Australia and how we're learning as well. Next slide, please. So I mentioned Australia is a little bit different. We're a federalised system of government. Our federal government, where I work, is responsible for national quarantine, international animal health issues, and the national coordination of disease control policies. Our state and territory governments, as in many federalised systems, are responsible for disease control, eradication, and animal welfare within their borders. But what's unique about Australia is that we have a not-for-profit publicly listed company called Animal Health Australia that's responsible for coordinating national programs, particularly national surveillance, farm-level biosecurity and emergency preparedness programs. It's co-owned by governments within Australia and also our peak industry bodies. And one of its main roles is to be the custodian of a cost-sharing deed, which sets out how we make decisions collectively between governments and industries and share the costs, obviously, of what we would call emergency animal disease responses. That's important because I'll talk about that in a little bit of detail later when I talk about performance standards. On to the next slide, please. So a little bit about Australia's context. We're about 1.7 times the size of the European Union, so a very, very large country. But we're the world's driest continent, apart from Antarctica. We've got very poor soils, and almost 20% of our land mass is desert. So despite our large size, we have reasonably small or low densities of livestock populations. Just to give you a bit of an understanding of this, we have 23 million beef cattle, about 2.6 million dairy cattle, just under 70 million sheep, and about 2.5 million pigs on the ground at any one time. One of the fortunate things about being an Australian is we don't experience transboundary disease incursions very frequently, but this in itself poses a problem for us because we don't really know how diseases would spread within our livestock populations, under our farming systems, and also under our environmental conditions. Next slide, please. So the sorts of public policy questions that the Australian Animal Health Services deal with are things like, well, what might be the epidemiological and economic impacts of disease outbreaks? How effective are our early detection systems? Are our contingency plans effective? What's our diagnostic testing capability or capacity in emergencies? And how do we measure performance of our emergency preparedness? Next slide, please. So identifying and securing resources and measuring our performance is quite a challenging issue for us. And I don't think Australia is alone with some of these problems. So this year, my colleagues and I published quite a simple policy framework to help address some of these problems. So it has 4 steps. The first step is assessing the nature of the risks that you want to control and identifying your response objectives. The second step is identifying the types of resources that would be required to achieve your desired objectives. The third step is to assess the available resources on the ground and identify any gaps. And the fourth is to make arrangements to assure the availability of resources in a crisis. And so in this paper, we're drawing some examples from Australia and New Zealand. Next slide, please. So one of the key ways Australia's approached these sorts of problems, as I mentioned earlier, is through epidemiological modelling. And Graham, through his team over just over 2 decades, has developed a long history in this regard, and it's something we're continuing within the Commonwealth and within the states and territories as well. Over that time, we've developed 2 major modelling platforms, one called AusSpread, which I'll talk about in a little bit later, and that was developed further into the Australian Animal Disease Simulation Model. Most of our modelling efforts have been focused on foot and mouth disease, being our most important risk as we see it, particularly because of the large consequences for an exporting nation. And over the years, we've used modelling for contingency planning, development, surveillance and resourcing assessments, as I'll discuss today, assessing the risks of feral pigs, the potential role of vaccination, potential animal welfare effects of our movement controls, understanding the economic impacts that might be directly and indirectly caused by foot and mouth disease incursion. But it's not only been foot and mouth disease. We've developed capacity to model classical swine fever, substantial body of work on avian influenza modelling, a lot of work's underway on African swine fever modelling, as is the case in many countries, and also pathogens like equine influenza, blue tongue, and TSEs. So next slide, please. So the first case study. So this case study really occurred after a review of Australia's preparedness for foot and mouth disease, which happened in 2011, which concluded that Australia might not be well prepared to respond to a medium to large outbreak of foot and mouth disease. That led to a detailed assessment of the resources that were available to respond in state and territory governments and industries, and that was used as the basis for some simulation modelling work. This work considered resource constraints for some key activities in disease control, so surveillance to identify infected premises, valuation of livestock on infected premises, and the resources available that would be required for stamping out on infected premises. And it simulated control with, under 3 scenarios. The first with unlimited resources to estimate what the ideal situation might be and what the ideal number of human resources would be required to respond optimally to an outbreak. A scenario where we looked at the available resources at the time, and also 2 situations with increased resources. So on to the next slide, please. So this might be a little bit difficult to see, and I apologise if that's the case, particularly for those of you on a mobile phone. This is just an example of some of the outputs, looking at the duration of the epidemic on the y-axis, and on the x-axis, you'll see labels for each of the resources. If we start on the far left-hand side, which is for baseline resources, at 6 months after an incursion, 40% of outbreaks were still ongoing with the current level of resources at the time, and 84% of outbreaks were ongoing at 3 months. With the resource level 1 scenario, which was assumed to be an additional 40 staff available between day 25 and 42 of an outbreak, and 80 staff after, all outbreaks were controlled within 6 months, and the longest duration of those being about 130 days. And 64% of outbreaks were controlled within 3 months, excuse me. With a high level of resources, so 100 staff available between day 25 and 42, and 200 staff available after, we didn't see much more of an impact. So whilst all outbreaks were still controlled within 6 months, and the longest duration was about 2 weeks less at 115 days. And 86% of outbreaks were controlled within the 3-month period. And on the far right-hand side, you can see with unconstrained resources, this estimated that our field operations would require over 2,500 staff. All of these outbreaks obviously were controlled within 6 months, and 98% were controlled within 3 months. So this allowed us to make some conclusions about the numbers of resources that we would require quite quickly in a response. Next slide, please. So I mentioned I'd talk about a functional simulation exercise that occurred in 2007 for our National Reference Laboratory. This was a functional exercise for highly pathogenic avian influenza. And one of the nice things about this exercise, in a strange way, was it occurred 2 months before our equine influenza outbreak. And so it allowed the laboratory to put in place some of the recommendations that were found. The objectives of the exercise were to test the laboratory capacity in an emergency, to investigate its client relationships, and also test operational management as well. So next slide, please. So this used a well-known simulation or scenario modeling system called the EmergoTrain system. And whilst that sounds very sophisticated, it is a proprietary system, but it's actually quite simple. It's used quite commonly actually in human medicine, particularly for mass casualty events, where fixed resources like the number of paramedics or the number of ambulances will really affect the outcome. I mentioned it was simple, and it involves the use of magnets that you can see on the bottom left of the screen that represent each of the resources that are available at the time. So the people obviously represent the staff of the laboratory and on a one-to-one basis. The test tubes represent an accession to the laboratory, and which was also recorded on a piece of paper. And those pieces of paper were moved between stations on whiteboards in the system, so it didn't actually use the real laboratory suites. And those represented the accessions area, the pass-through box, the isolation rooms, the PCR lab, the RNA extraction area, and real-time PCR and reporting. What probably added to the stress of the exercise was it occurred at about 4 times the normal speed. So there was a clock on the wall running at an hour every 15 minutes, and that probably added quite a bit to the stress for the individual participants. But I mentioned it was very useful. The outcomes were they made some recommendations around personnel management and capability, particularly liaison between the research side of the laboratory and the diagnostic side of the laboratory, It identified some issues around equipment and reagents at the time. It also identified the need for some financial planning as well, which is also fundamentally important for preparedness, in my opinion, and led to the development of a process modeling capability for diagnostics within the laboratory. Next slide, please. So on to performance measurement. Perhaps not some of our favourite topics, but I think it's critically important to assure our systems are effective. I mentioned before our Emergency Animal Disease Response Agreement in Australia, which I'll just repeat is a cost-sharing deed between governments and industries that sets out the responsibilities for decision-making and also the sharing of costs. But one other thing it does is it sets out the so-called normal commitments or performance requirements for each of the parties under the deed. So for government parties, I think you won't be surprised to know that it covers things like the capacity for early detection, the capacity to investigate a suspected outbreak, diagnostic capacity, and also response capacity. But there's also responsibilities for industries too. That includes things like having qualified personnel for the decision-making process, being able to meet the cost-sharing responsibilities, to implement risk mitigation at the industry level, to publish biosecurity plans, and to support compensation, communications, and also provide advice on animal welfare. So there's a link at the bottom of the screen that provides you with some more detail about those sorts of qualitative performance metrics. To assess our capabilities underneath these normal commitments, we undertook a national assessment through the Performance of Veterinary Services tool in 2015, and that led to the development of a state and territory self-assessment tool for governments. There's also some work underway to benchmark industry's capabilities using a similar tool as well. Next slide, please. So one of the heroic things that Australia's committed to do in 2021 is to develop performance measurements for its entire biosecurity system. So that's covering the anticipation, prevention, screening at the border, emergency preparedness, detection, response, and recovery capabilities for not just terrestrial animal health, but also aquatic animal health, plant health, and also environmental pests and diseases. So it's a huge task. We're trialling a maturity matrix approach for this task, and this is an example of one that we've used in the past for this. On the y-axis, there are different categories and criteria that an institution would like to be assessed against, and just reading those out, they're planning, resourcing, training and education, exercising, and evaluation and corrective action. And on the x-axis, there's an ordinal scale, which represents the different assessed categories. The first is improvised, the second is developing, the third is evolving, and the fourth is optimized. Just if you could click on the next animation, please. And one more, please. Thank you. This just zooms in on what the improvised capabilities were assessed as in this maturity matrix, which was having capabilities and capacity being evaluated in real or detailed simulation responses. Next slide, please. So we proposed quite a provocative question at the start, which is how much is enough? And I haven't answered that question. I haven't answered it deliberately because I think it's a value judgment. I think it's a value judgment based on the risks that a country finds itself with, their own context and industry requirements, and also the resources available. So I provided you with some quick examples of how we've used epidemiological modelling, simulation exercises, and qualitative performance metrics within Australia to assess current and desired performance. And I hope it's been useful for you to answer these questions that are difficult to answer with science alone. Thank you very much for your attention.
Thank you very much for this presentation, Sam. Very interesting. I was thinking, how have you involved the private sector in all of this planning and getting prepared for control? for these outbreaks. I know that sometimes the private sector has a different opinion of how things should be done, etc., etc. Have you taken that on board in your program, and how do you do that? And how do you work with the private sector in all this also to finance it?
There's some very big questions, Sten, and I'll try and answer each one in turn, if I may. I think when we talk about the private sector, we need to be very mindful of how diverse it is, particularly even in a country so big as Australia. Often our governments will interact with our peak industry bodies, so statutory organisations that are funded by levies, through mechanisms like the Animal Health Australia mechanism that I mentioned, which allows for co-funding between each of the shareholders. But I think one of the big challenges within the Australian context, particularly when infectious diseases aren't major limitations to farm productivity, it's a big challenge to ensure that biosecurity and infectious diseases remain in the forefront of farmers' minds. And what I mean by that is, well, we undertook some behavioural analysis of farmers probably about 3 or 4 years ago now. And one of the key findings was that they're much more concerned about things like climate variability, much more concerned about price variability. And I dare say at the moment with trade and market access issues and issues along the supply chain as well, with abattoirs closing in Australia in the past. So it's a big challenge and one that we do need to be mindful of. And I think into the future, organisations will look at more using electronic means, using social media, and using directed campaigns to try and raise that awareness. So I hope I partially answered your question, Sten.
Well, yes, you did. Thank you very much, Sam. And then I think we will move to the next presentation. And Carissa, please introduce.
Certainly. Hi. Well, thanks firstly to Sam for a great presentation. And just a reminder to everyone, if you have any questions, any further questions for Sam, that you can post those on the forum, and we'll get to as many questions as we can at the end of this session in the roundtable. So our next keynote speaker for today is Donald Salmon, Director of Laboratory Services of the Department of Agriculture, Food and the Marine. And Donald will be presenting on diagnostic supply chain issues, lessons from COVID-19 for animal health. Welcome, Donald.
Thanks, Carissa. And good morning, everybody from Ireland. Some of us who are ordinarily engaged in animal health-related testing in Ireland have been quite involved since the beginning of this year in assisting our public health and medical counterparts in testing different aspects of testing for COVID-19 or SARS-CoV-2. And I suppose that was an instructive experience, and there are lessons perhaps we could take from that for dealing with epizootic disease. Next slide, please. So this unusual manifestation, if you like, of One Health approach was not unique to Ireland by any means, and so much so that OIE published helpful guidance for veterinary laboratories across the world that were engaged in this kind of work earlier this year. Next slide. The testing landscape for COVID-19 in Ireland, well, ordinarily, our National Virus Reference Laboratory would deal with pretty much all of the requirements of human healthcare in terms of viral diagnostics. So that's an entity of about 90 people. It's based on a university campus in Dublin. But their capacity was quickly overwhelmed by the requirements of the pandemic. So the testing, if you like, then extended out to our acute hospitals, which have PCR testing capability. Then we became involved, our Department of Agriculture laboratories. So we have a range of functions that we discharge, national reference laboratory and diagnostic functions across animal health, plant health and food safety, for which we make quite significant use of PCR testing. And last but not least, a private commercial laboratory that's ordinarily engaged in animal health related work, which is the single largest provider of PCR testing in the state for reasons that I'll come back to later. Next slide, please. So the National Virus Reference Laboratory, or NVRL, were tasked with developing testing capability for this new virus, this new disease. And like everybody else, they were able to avail of sequence data provided by the Chinese Center for Disease Control. And 10 days later, a protocol that the World Health Organization published for an RT-PCR assay. And this is quite a robust assay. It had two gene targets, an envelope gene, which is common to the SARS family of viruses, and a polymerase gene specific to the SARS-CoV-2 virus. They didn't actually have control material specific for SARS-CoV-2 for about a month after that, when it was provided by our UK counterparts, or their UK counterparts. And the first case that was confirmed in Ireland was at the end of February of this year. And around about that time, end of February, beginning of March, the first PCR test kit became commercially available. So next slide, please. So the capacity of NVRL was very quickly overwhelmed. They validated this new commercially available test kit. They made it available to hospital laboratories. At least 11 of those hospitals immediately adopted the test, and this was largely used for the purposes of inpatients and healthcare workers in those hospitals. We were able to step in at that point because one of the limiting factors immediately for NVRL was having the space and the people to deal with these onerous sample handling, virus inactivation, and data entry steps at the beginning of the testing process. So we were able to provide space and mobilize people, which effectively helped them to double their test capacity. But our public health authorities predicted that we would need to get up to at least 10,000 tests a day very shortly, which proved to be the case. And that required us to bring on board ENFR with their mass testing pipeline. And around about that time, and a major constraint on developing testing capacity worldwide, was a shortage of this critical component, GITC, and lysis and extraction buffers, accentuated in cases of people requiring particular components or consumables containing this for closed system extraction platforms. Next slide, please. So our hospital laboratories, on top of everything else, on what happened to be Friday the 13th of March, were told that this PCR test kit that they had adapted was no longer available. So they had to immediately turn around, source alternatives and validate those for use. They also had a huge reliance on one particular automated closed system extraction platform, so a market leader in Ireland at any rate, but I suspect in many other European countries. And they had to source and validate alternative platforms and alternative sources of the reagents used in those platforms or on those platforms. They entered into contracts for the supply of instrumentation on the understanding and given a guarantee that they would also be supplied with test kits to test a certain minimum number of samples. So they signed the contracts, the instruments were placed, but lo and behold, the test kits didn't arrive, or certainly not as many as were promised. And I think that's something we'll come back to in terms of the lessons we can take from this. And last but not least, one I think very successful innovation and speaking with people across these hospital laboratories. So our acute hospitals and the laboratories embedded within them are quite independent entities. They don't have a nationwide management or coordination system. So it was very important that somebody stepped up here and convened weekly Zoom calls as it happened. And people very quickly started sharing information. They started learning from each other. They avoided making the same mistakes. They avoided replicating efforts. And they were able to greatly speed up the time taken for validation of different test kits and platforms. They were able to quickly identify which test kits worked with which extraction systems. And in some instances, they were able to provide feedback back to manufacturers who were not aware of some of the limiting issues here. Next slide, please. So our department became involved, as I mentioned, primarily in the first instance to assist NVRL in sample handling and in data entry. So we were able to provide a biocontainment laboratory in which we had 8 biosafety cabinets where people could work safely with human specimens, inactivate the virus, and then we returned the virus to, returned the samples rather, to NVRL for testing. And we also entered data that came in handwritten forms onto their LIMS system. Somewhat clunky, but bear in mind that at that time we were also struggling with the same issue around availability of reagents for extraction. And this also impacted our more routine non-COVID work. So we were contemporaneously dealing with an avian influenza outbreak, and we also had important official testing to do for import, for export, for other purposes, where we wanted to rely on our accredited methods using that particular extraction system. Next slide. So I mentioned EnFIR laboratories, and they have pretty huge PCR testing capacity because of the success of this national BVD, bovine viral diarrhea eradication program that we've had in Ireland for the last several years. So 2.5 million calves are born in Ireland every year. They have to be tagged and registered by the owner. One of those tags takes a plug of tissue, and the owner is obliged to send that to one of 7 laboratories that are designated for testing. Next slide, please. So EnFIR laboratories would have the lion's share of that market. They typically test about 60% of those ear tag tissue samples. And you can see here from this graph shows the number of calves born by week in Ireland. This is the data for 2019, but similar for just about any other year. And you can see a huge peak in springtime. So week 7 here towards the end of April, 170,000 calves registered and having to be tested. So EnFIR, next slide please. EnFIR have a peak or surge testing capacity of up to 100,000 PCR tests for BVD a week. The system they were using for that, it's a relatively crude extraction system to be able to get access to BVD virus in ear tissue. It wasn't considered appropriate for testing for SARS-CoV-2 in swab samples. So they had to look around and source an alternative system in turning their pipeline around to COVID-19 testing purpose. They were also trying to contend with this issue around the shortage of particular extraction buffers. So they looked to source an extraction system that they knew wasn't being used elsewhere in Ireland, a system that came from the US, and a system that was an open system that could work with most of the reagents supplied on the market. They also deferred back to the World Health Organization's assay using materials that were developed and controlled for them by the National Virus Reference Laboratory. So they were able to very quickly scale up to testing about 10,000 samples a day. The overall sampling and testing system, so EnFIR would have primarily dealt with samples coming from community testing hubs across the country. And if you look at that overall system of community sampling and testing, there were several sequential bottlenecks in supplies of the materials needed for that. It started out with PPE for the workers, then it was this issue with the extraction buffers, then it was swabs to take the samples, then it was plasticware for the different machinery that's used here for the extraction platforms and the PCR testing platforms. And last but not least, as recently as this week, laboratory scientists in mass testing laboratories across the world are writing to Santa looking for 50 microliter filter tips. Next slide, please. So as time moves on and as supply and demand come closer into sync, one problem begins to abate a little, but a different type of supply chain challenge becomes apparent. So very large numbers of rapid testing kits have come to market over the last few months. The World Health Organization, in their guidance issued in September, mentioned that they were aware of nearly 100 companies that were producing rapid tests, but very few of these were subject to any form of stringent regulatory review. That does not stop the companies that have produced these from engaging in intense extensive marketing and lobbying campaigns. So you end up with quite significant public and political pressure to deploy rapid tests, and that has certainly been the case in Ireland. And next slide, please. So it is important that, and we do have, more or less, we have established processes, or we know how to go about this, how to look at comparative evaluation and validation of test methods. Our public health authorities went to market. They recently had more than 50 commercial offerings of rapid tests. So that represents a very significant amount of work, initial desktop evaluation of the information provided by the manufacturers, wet lab verification of some of that data. And we came in and we became involved, and we are currently involved. We have a team of laboratory scientists today involved in a field evaluation exercise in one of our meat processing plants. So like many countries across the world, we've had a particular problem with COVID-19 outbreaks earlier this year in our meat processing plants. And we have invested significantly in surveillance and testing in meat processing plants. So whilst the evaluation and validation strategies might be quite clear, testing strategies aren't quite as clear cut. Who is going to do these tests? When will they be used? How will they be done? Who will have first access to information? And the $6 million question, what do we do when we get a positive? Next slide, please. So what are the lessons that we in veterinary laboratories and potentially having to deal with epizootics in the years to come might take from this? So my little diagrams of, and I'm thinking of Sam's magnets here, these little diagrams of factories and trucks are a crude attempt of mine to kind of show the journey that many of us have been on over the last few decades. We have moved from systems where a lot of us bought reagents and materials from various different sources. We cooked up most of our own assays in-house, and that gave us considerable flexibility. But as we have embraced accreditation, we have essentially outsourced quality control and batch control as we've become increasingly reliant on commercially available test kits. But that has brought with it a certain vulnerability. There's a trade-off there, that our supply lines have narrowed, and we've become very dependent on particular systems, particular suppliers, and any problem there, and we have a problem. So what might we do? We could look at stockpiling, at increasing our inventory, but a lot of these materials have fairly short shelf lives, and it's not really practical to think of stockpiling every single reagent or test kit we use, because for one thing, we don't actually know what the next big thing is going to be. The second thing that we sometimes think of is contracts for resupply of materials. But as we've seen, commercial companies, they may promise something in good faith, but when push comes to shove, I suppose hard commercial realities kick in, and they may think, well, better to have a whole load of partially satisfied customers than a small number of very satisfied customers and everybody else entirely dissatisfied. So as that great philosopher Father Ted might say, down with that sort of thing. But maybe more pragmatically, you know, we should think, well, look, that's the way they're thinking about this. They're also thinking, for me to increase supply here, I probably have to invest hugely in a new production line, make a big capital investment, and by the time I've done all that, maybe the same demand won't exist. So the secret in all of this is, I think, and it's easier said than done, that we need to look at diversifying the systems and supplies we use and the suppliers we obtain those from. Next slide, please. Flowing from that, we need to be more agile, more adaptable. So we need to be ready to embrace new materials that come to market, alternative approaches, for example, with PCR, the possibility of pooling to make more efficient use of the materials we have, and to look at new and alternative testing systems. And the key to all of this is being able to quickly validate any of these new approaches and those keywords as fit for purpose, that they do what we need them to do. Next slide, please. A third lesson, excuse me. A third lesson is that we communicate and collaborate with our peers. So I think it's instructive here that at international level, the speed with which information was shared, protocols were developed and shared, and people were able to develop capability. But also at national level, that we don't replicate the same errors, we don't make the same mistakes, and that we more efficiently allocate resources and materials and samples. So we make the best possible use of all of the resources we have. Next slide. And when it comes to the think in terms of when we're contingency planning, not just things as we expect them to be or our system as normal, but think of what if scenarios. What if we didn't have PCR testing? What if we were to apply rapid testing? Those of us who remember EuFMD in 2001, remember the debates about pen-side tests then, and many of the same questions were raised then as have been raised in recent months about rapid tests for COVID-19. Last but not least, I'll leave you with a thought. It has nothing really to do with supply lines, but it was an instructive experience from working with our medical counterparts, and maybe an inherent advantage that we sometimes take for granted. Our medical and public health counterparts have a very difficult space to navigate in dealing with this outbreak. There's a huge focus on individual patient care, on the autonomy of the person, on privacy of data, on medical ethics, and on medical legal considerations. So it's very hard for people to make that conceptual leap from dealing with the individual to dealing with populations. Those of us from an animal health perspective, our default is to think of populations from policymakers through to field investigators. And that's an enormous advantage when you're trying to deal with a highly infectious disease. And my last slide. So to thank my co-authors in this paper, Maura McElroy, Jeff Connell, Susie Coughlin, and Mary Keoghan. And the picture here, the whirling dervishes, the people who are more than 2 meters apart, represent all of our laboratory colleagues across all of these different entities, and the marvelous work that they have done over the last few months. Thank you very much.
Thank you very much, Donald, for a very interesting presentation. A veterinary lab getting into testing of human samples, working together with the human health labs. It's approaching One Health, right? And I think you were, in the final part, you were a little bit pointing to what's the difference. The difference being populations is the veterinary focus, whereas the individual in humans. But can you say a few words more about, did you have any problems with the differences in culture between human health and veterinary animal health? A difference in technical language, or a difference in expectations of what a lab should do? Can you expand a little bit upon that? Because I think that's very interesting.
Yeah, thanks, Sten. I think where that became most evident to us has been over the recent weeks, when we have been involved in field evaluation of these rapid tests in meat plants. And I suppose there's a brutal simplicity to the way we approach herd health and disease investigation in veterinary medicine. And sometimes that doesn't sit very well with those who come from a hospital setting. And you know, we also have to be conscious of the fact that you're dealing all of the time here with a human subject, you know, somebody who, you know, is emotively engaged in what their result is going to be and has their own concerns, maybe, you know, people at home who are they're concerned about. And so that was a learning for us. But I think we also brought a certain pragmatism to the table when it came to moving on with how these tests are evaluated, that there might have been a touch of a council of perfection in terms of how this was being done. And I suppose I'd be an advocate of, a good plan today is better than a better plan at some point in the future.
Well, thank you very much. I look forward to the discussions in the roundtable later on. And for now, we will need to move on. And Carissa, please, can you introduce the next speaker? Thank you.
Great, thank you so much. Thanks again to our two keynote speakers, Donald and Sam. We're getting some really good questions through the forum, but please do keep them coming so that we can make sure that we have a lot of questions to answer for you during the roundtable at the end of this session. So we now have 4 recorded presentations, after which we will have the live roundtable. Our first presenter is Nagendra Singanallur from the Commonwealth Scientific and Industrial Research Organisation, or CSIRO. And Nagendra will be presenting on systems immunology-based approach to study early immune responses to emergency foot and mouth disease vaccination in pigs. Thank you.
Good afternoon, everyone. I'm here to present some preliminary results on using a systems immunology approach to assess early immune responses to intradermal vaccines in pigs. Pigs respond poorly to EuFMD vaccines, and the antibody titers are often low and do not correlate with protection. Reports from South Korea in 2018 raised concerns on carcass quality post-vaccination, with abscess formation and granulomas reported from the site of vaccination. This resulted in poor vaccine uptake, leading to inadequate implementation of the control programs. Ablay and co-workers in 2009 have shown that 1/10 of the regular vaccine administered by intradermal route was effective and protected against challenge in pigs. The antigen concentration in such formulations can be reduced along with dose volume, thereby reducing the cost of vaccines and vaccination, and also reduce the adverse effects in pigs. Several preliminary studies were carried out to identify suitable adjuvants that can deliver EuFMD vaccines by intradermal route. The antibody response and homologous protection were compared with the intramuscular vaccines. Based on these studies on different adjuvants, one promising candidate was selected. The vaccine formulation with the selected adjuvant was comparable with intramuscular vaccines in a homologous challenge study. We observed small differences in the antibody titers between the intradermal and intramuscular vaccine groups. Therefore, a pilot study was carried out to investigate the use of systems immunology-based approach to look into the early immune responses in pigs to intradermal vaccines. 2 groups of 8 pigs were vaccinated either with an experimental intramuscular vaccine or an intradermal vaccine. Intramuscular vaccine 2 ml dose, intradermal vaccine 0.2 ml dose. 5 control pigs were used. They were unvaccinated. The antigen concentration in both the vaccines were similar. Potassium ED2 blood was collected on day -3 and 3 post-vaccination for systems immunology studies. PBMCs were separated from the whole blood. Total RNA was extracted. The quality control check was performed on RNA concentration and integrity, followed by TrueSeq stranded mRNA protocol for preparing RNA-seq libraries. Next-gen sequencing was carried out using NovaSeq 6000 systems Illumina platform to obtain at least 20 million paired sequence rates per sample. A standard bioinformatics pipeline was followed to determine the differential gene expressions post-vaccination into 2 vaccine groups. Gene set enrichment analysis was performed using predefined human blood transcriptome modules based on published literature. Following DSeq analysis, genes with positive or negative log2 fold change of 1 combined with an adjusted p-value, also referred to as false discovery rate of less than 0.05, were represented as volcano plots showing the up and down regulated genes. Since groups of genes often act as networks, have an interplay and downstream knock-on effects on each other, it is expected that some genes would be upregulated while the others downregulated even within the same pathways. In these volcano plots, the upregulated genes are shown in green, while the downregulated genes are shown in red. We have also highlighted few selected genes that are known to play significant role in the innate and adaptive immune responses. In the unvaccinated control groups, there was no differentially expressed genes on days corresponding to 3 and -3 post-vaccination. 2 of the 5 control pigs with underlying adverse health issues like weight loss and abscess development were excluded from the analysis as outliers. In pigs that received the intramuscular vaccine, 177 genes were upregulated, while 43 were downregulated. In the intradermal group, 138 genes were upregulated, while 21 were downregulated. Overall, it appeared that the transcriptomic response was less pronounced following intradermal vaccination compared to intramuscular vaccination in terms of the number of genes up or down regulated. However, we identified some overlap in specific groups of genes between the 2 vaccine groups, but also differences where regulation of certain gene was dominant in the intradermal group compared to the intramuscular vaccine group. Many genes showed significant up and down regulation pre and post vaccination. Here we are showing the change in expression of 2 specific genes that target different responses in pigs, the inflammatory response and the dendritic cell antigen presentation response as per plots. The blue color represents the control group, red the intradermal, and green the intramuscular groups. Note that this represents a comparison of gene expression within each group pre and post vaccination and not between the different routes of administration. ATF3 is an activating transcription factor involved in inflammatory response due to cellular stress and drives the innate immunity. It is a negative regulator of TLR signaling in macrophages and dendritic cells, thereby dampening expression of pro-inflammatory genes and preventing overreaction of the immune system. GPR157 is a G protein coupled receptor that are on the cell surface to detect molecules outside a cell and activate cellular responses. These receptors are known to play a role in development and activation of the macrophages. The post-vaccination levels were highly significant. Genes can be grouped into specific families based on common function that direct specific parts of a complex immune response network. These are referred to as blood transcriptome modules or BTM families. A clearer understanding of the overall effect of the treatment or vaccination can be understood using these modules in a gene set enrichment analysis. Both intramuscular and intradermal vaccination positively modulated the BTMs related to natural killer cells and T cell surface differentiation, activation, signaling, co-stimulation, and proliferation. It was more pronounced in the intramuscular group compared to the intradermal group. Similarly, the BTMs involved in innate and inflammatory responses were positively modulated by both vaccine groups, but to a greater extent in the intradermal vaccine group compared to the intramuscular vaccine group. Blood transcriptomic modules involved in B cell activation and differentiation, as well as B cell receptor signaling, were negatively modulated in both vaccine routes. Gene networks involved in cell division of stimulated CD4 helper T cells that are responsible for activating B cells as part of adaptive immune response were also negatively modulated. In summary, we could demonstrate significant up and down regulation of several genes following intramuscular and intradermal vaccinations 3 days post-vaccination. Several statistically significant individual genes involved in immunity were identified. Positive modulation was observed in gene sets targeting natural killer cells and T cells. Negative modulation was observed in gene sets targeting B cell responses and helper T cells. Transcriptomic profiles were more likely influenced by adjuvant formulations, as adjuvants are designed to derive specific types of immune responses. PTM analysis also suggested that both vaccine routes drove the immune response towards innate and cellular immunity. In conclusion, we have demonstrated that a systems immunology-based approach can be used to assess the early immune responses to EuFMD vaccines in pigs. The transcriptomic analysis in this pilot study showed subtle differences in early immune response between intramuscular and intradermal routes. Similar analysis at different days post-vaccination could provide more evidence as to why pigs generate low humoral response in general. The next step in these studies are to directly compare the responses between intramuscular and intradermal groups. Further studies to compare prime only with prime boost vaccination and validate these results using a vaccine efficacy study in pigs. Similar transcriptomic studies are needed to evaluate stimulation of favorable responses by specific adjuvant formulations. Thank you very much.
Thanks so much to Nagendra for providing that fantastic presentation. I'm sure we'll have some good questions for you come the round table, Nagendra. So next up, we have a recorded presentation from Sarah Watson. Sarah is currently with the European Commission for the Control of Foot and Mouth Disease, and she will be presenting on developing an analytical model to predict FMD vaccine demand for endemic countries. Thank you very much, Sarah.
My name is Sarah and I'm an intern for EuFMD under Pillar 3. I am a recently qualified vet with a Master's in Control of Infectious Diseases. Alongside my supervisors, Paolo, Buda and Nick, we are developing an analytical model to help predict foot and mouth disease vaccine demand for endemic countries. It's important to predict FMD vaccine demand. As the global human population continues to rise, livestock numbers are set to follow. There is therefore an ever-increasing foot and mouth susceptible population. 75% of FMD is located in areas with the lowest income per capita. Vaccination is a key component of endemic countries' control strategies. However, only around 7% of the global vaccine market is reaching them. Vaccine production is an investment to manufacture, but also to purchase. Often this means there is a supply-demand mismatch. Predicting accurate dose numbers is difficult due to the challenge of forecasting FMD prevalence and uncertainty of the proportion of livestock targeted with vaccination. The model was developed to highlight further the vaccine demand mismatch. It aims to provide key stakeholders with a vaccine dose estimate for a country according to their control strategy for the consecutive year. This will hopefully inform not only countries but manufacturers, researchers and donors of the extent of the vaccine deficit. We also believe it could be used as a tool for countries to benchmark against a total vaccine estimate versus what is actually being used on the ground. This will hopefully identify areas in their control plans which need improving. It will also, towards the end of its development, predict global vaccine doses. This, we hope, will highlight the extent of the FMD vaccine inequality problem. The model is in its early development stages currently, so it is relatively simple. This is to allow for flexibility in its development further on. There are two sides to the model: emergency vaccination, which is reactive vaccination in response to an outbreak, and prophylactic vaccination, which is vaccination as part of a country's control programme. The control strategy is linked to each country's progressive control pathway stage as developed by EuFMD. Prophylactic vaccination, as seen in the diagram, first starts by estimating the next year's livestock population. The population is then split into young stock and adult stock to allow for the variation in vaccine doses. Young stock are considered to be below 12 months of age. They typically have a primary course of 2 doses 1 month apart and a booster 6 months later. Adult stock are usually on 2 doses a year, but will depend on the country's control programme. The PCP stage then suggests what proportion of the livestock population are prophylactically vaccinated. Emergency vaccination requires forecasting of FMD outbreak numbers. This is calculated by multiplying previous year outbreak numbers by a predicted reduction percent based on a country's PCP stage. Emergency vaccination occurs in a set area surrounding an outbreak. The size of this area is country dependent. The livestock population in the area is calculated based on livestock density. Not all outbreaks are typically covered with emergency vaccination, and so a coverage percent is again estimated via PCP stage. The model has been developed using R software, and due to the uncertainty and variation surrounding many of the parameters, a stochastic approach was taken. A Monte Carlo simulation with 1,000 iterations of the model will occur during its final stages of development. There are several key parameters in the model. Population growth was calculated using the Food and Agricultural Future of Food and Farming report. It was published in 2018 and estimates livestock population growth along with other agricultural variables from 2020 to 2050. It states that there are 3 possible scenarios for the future of agriculture, and this provided the range for our growth distribution. Prophylactic vaccine coverage percent, outbreak number reduction percent, and outbreak vaccine coverage percent was estimated based on the PCP stage the country is in, informed through an expert elicitation survey. Experts in the field were asked to assign proportions to each of the three livestock categories: large ruminants, small ruminants, and pigs. This is discussed in more detail in the following slides. Livestock density was simply the livestock population divided by the surface area of that country. It assumes that livestock distribution is equal. Further development of the model will include within-country variation of animal density, particularly since outbreaks may be more likely to occur in more animal-dense areas, so that this will likely underestimate demand. As both emergency and prophylactic vaccinations are in separate sections of the model, it assumes that those livestock which are vaccinated in an outbreak continue their routine vaccination as normal. In reality, there may be some country-to-country variation with regards to this. The data inputs for the model came from several sources. Data on population and outbreak numbers was from the OIE-WAHIS interface. We found this to be the most comprehensive database for FMD outbreaks. However, we are aware that not all countries will be able to submit accurate information to this platform. It therefore has its own limitations. The PCP stage projections were from roadmap meetings carried out as part of the PCP. They are generated from each country's predicted progression through the PCP, based on several factors, including current and historic activities towards FMD control. The countries not in the PCP are currently not included due to several parameters which rely on the PCP staging. As mentioned on the previous slides, parameters linked to the PCP stages were from an expert elicitation survey. An expert elicitation survey was produced to assign a range of values to the parameters linked to PCP stage. It was sent out to around 80 experts in the field via email. Roughly 20% completed the survey in round 1. The experts were also asked to nominate others who they believe would be able to provide insight into the topic. The three main parameters requested in the questionnaire related to the number of outbreaks which would be expected to produce each year if a country was either to remain in a PCP stage or progress to a PCP stage, i.e. this is the percentage reduction in outbreaks per year you would expect if the country was to stay in PCP 1 or to move from PCP stage 1 to 2, etc. The proportion of outbreaks covered with emergency vaccination for each PCP stage, i.e. if the country was in PCP stage 1, how many outbreaks would you expect to be covered by emergency vaccination? And finally, the last proportion is the proportion of livestock which are routinely vaccinated as part of a country control programme for each PCP stage. For example, a country in PCP stage 1 may only vaccinate 40% of the resident cattle population, 20% of sheep and no pigs. The survey results were weighted depending on the candidate's own opinion of their expertise on FMD and the PCP. Unfamiliar, casually acquainted, familiar, knowledgeable and expert. They were then asked for how many countries this was relevant for, i.e. did they feel they were experts in a single country, regionally, which is multiple countries in the same region, internationally, multiple countries in more than one region, or globally, multiple countries in several regions. We used the linear opinion pool method to weight the responses from the experts, as highlighted in the diagram. We assigned beta-PERT distributions to the responses and combined that with each expert's weighting. All the distributions were combined using model risk from Vose software on Excel. The mean and standard deviations were extracted from the final combined distribution. Results were variable between the experts, which meant standard deviations were large. However, a common trend was that as countries moved through the PCP, the percentage of livestock population vaccinated prophylactically would increase, and the number of outbreaks targeted with emergency vaccination would also increase, particularly in stage 4, which is the final stage before OIE recognition of freedom with vaccination. This is highlighted in Figure 1, which depicts the mean vaccination coverage percent per livestock category by PCP stage. Interestingly, PCP 3 and PCP 4 seem to have a similar percentage coverage. This trend also occurs in Figure 2, where the proportion of large ruminant outbreaks targeted remains similar from PCP 3 to 4. Contrasting expert opinion occurred with regards to the proportion reduction of outbreaks per PCP stage. The most common problem was that many didn't believe PCP stage was a reliable way of predicting outbreaks, especially during the early stages. Further comments on this will be made in the next slide. Each expert was given the opportunity to comment in each section of the survey. Many experts believe that an association between the incidence of FMD and a country's PCP stage is weaker during the early stages, 1 to 2, as there are no strict control strategies being implemented, compared to stages 3 and 4. Additionally, FMD incidence is variable per year regardless of PCP stages, which makes the prediction of outbreak reduction challenging. Lots of countries also target certain areas for higher vaccination coverage, which means their control strategies and PCP stages vary within the same country. Finally, many believe that emergency vaccination should not occur in PCP stages 1 and 2, as it is not commonly part of the control plan at this point. These comments have been taken on board and will be summarised for further comment in the next round of the expert elicitation. Further model development is still ongoing. Another 2 rounds of the expert elicitation will occur over the coming months, which should improve the accuracy of our PCP stage-related values. We are aiming to split each country into regions. This is useful for those who may have varying control strategies based on risk hotspots. It will also allow for more accurate emergency vaccination doses to be estimated, as livestock density will vary per region. We are going to split the large ruminant population into commercial dairy and beef, pastoralists and smallholders, as each section may have different preference on vaccination schedule and responses to outbreaks. For example, commercial dairy farms may wish to vaccinate more frequently than a smallholder. Equally, there may also be varying population dynamics in each sector. Commercial dairy and beef are more likely to have a larger young stock proportion compared to the smallholders and potentially a higher livestock density. Similarly, we are still developing the young stock and adult stock proportion splits to help with the variation in vaccine doses per year. Lastly, there will be ongoing model evaluation to ensure that values predicted are analysed for accuracy. In order to do this, we are looking to compare model predictions with individual country reports. We're going to complete sensitivity analysis for some of the variables in the model. This will hopefully identify which variables have a more significant impact on vaccine dose estimates. A big thank you to Paolo Budenick and the EuFMD team for their ongoing help and support. Thank you to everyone else who has been involved with the model so far. Thank you very much for listening. The model is in its early development stages, so we welcome any feedback or comments you may have. Thank you very much.
Thank you very much to Sarah for that presentation. It's really exciting to hear where this work is up to and the plans for the further model development as well. We now have our next recorded presentation. This is a joint presentation from Lisa Karoth from the who is the Emergency Program Manager at the California Department of Food and Agriculture. And she'll be joined by Pamela Hullinger from EuFMD. And neither of these presenters are going to be present with us at the roundtable, unfortunately, due to the time zone differences, but please do still post questions for them and they will answer them this afternoon when they join us. Thank you very much.
Welcome, and thank you to the EuFMD for the opportunity to share this short summary of the historical and ongoing work being done by the California Department of Food and Agriculture on foot and mouth disease vaccination contingency planning. My name is Pam Hullinger, and I'm a veterinary epidemiologist currently working for the EuFMD. My co-presenter, Lisa Kiros, leads the Emergency Preparedness and Response section of the Animal Health Branch at the California Department of Food and Agriculture. We are co-presenting as I was a collaborator in the early aspects of this work. We hope you find this presentation helpful in the context of your foot and mouth disease planning efforts. While we can't join you in real time for questions, we'd be happy to follow up via the meeting's chat feature. The goal of an emergency foot and mouth disease vaccination campaign is to suppress virus replication in the high-risk animals by rapidly being able to vaccinate a high proportion of the at-risk population. In California, the selected vaccination strategy and the disposition of vaccinates will depend upon the nature of the outbreak and the population at risk. California is a large state with a diverse livestock population. One of the greatest concerns is introduction of foot and mouth disease into the dairy-dense region of the California Central Valley. Disposition of vaccinates may include euthanasia and disposal, slaughter for consumption, or living out productive lives, or any combination of these options. While the USDA ultimately controls access to foot and mouth disease vaccine in the United States, the state, in coordination with USDA, is responsible for identifying the most optimal vaccine strategy and providing operational support to execute the vaccination campaign. California's early planning efforts were inspired by the successful use of responsive foot and mouth disease vaccination in Uruguay's national cattle herd in 2001. This vaccinate to live approach was successful in controlling the outbreak with minimal slaughter, utilizing 2 rounds of vaccination for the nation's cattle herd of just over 11 million head. So in 2010, in collaboration with Uruguay's Ministry of Agriculture, California learned valuable lessons about procurement, product testing, approach to vaccination distribution, cold chain maintenance, and the use of industry vaccinators with government oversight and monitoring. In particular, the animal identification system and movement tracking database in Uruguay were quite impressive. California has seen the need to devote time and resources to foot and mouth disease vaccination planning for several reasons. First, if an outbreak were to occur in the dairy-dense region of the state, the outbreak would have likely spread before detection due to the nature of animal and product movement in this highly concentrated area. Additionally, for dairies, indemnity is costly and the public tolerance for large-scale depopulation would be very low. The logistical challenges and environmental impacts of large-scale disposal are significant and present increased biosecurity risk in the face of limited rendering capacity. Planning efforts started back in 2010 and have included an exercise focused on receiving and redistributing the USDA National Veterinary Stockpile assets, the utilization of private veterinarians in vaccination delivery, foot and mouth disease controlled movement workshops, and most recently a full functional national exercise that explored bulk milk tank screening for foot and mouth disease, along with requesting FMD vaccine from the USDA. And now it is my pleasure to turn the talk over to my colleague Lisa Quiros, who will discuss in more detail California's planning considerations for foot and mouth disease vaccination.
Because vaccine will be manufactured to match our outbreak, we know there will be a delay between when we request vaccine from USDA and when the vaccine arrives in our state. If we believe there is a credible threat and we will use vaccine at all, it is important that we request vaccine early on, maybe even before there is an actual detection in our state. We've talked about triggers for requesting vaccines, such as detection in North America. So how do we know how much vaccine to request? Our state has a good estimate of animal populations based on several available data sets. But in order to know how much vaccine to distribute to each farm, more specific data will be needed. The USDA has developed a herd plan template that can be used to collect this farm-specific data. Our plan is to capture this information while we are waiting for vaccine to be delivered to our state. During this time, we will contact individual producers, have them sign the herd plan and agreement to vaccinate, and update and validate head counts on each premises. It's important to ensure long-term strategy planning along with immediate response. Someone needs to plan for what will be required to regain freedom from disease status from the very beginning. Once the vaccine is requested, if staffing is limited, many of our responders may be busy with other critical response activities. It may be necessary to assemble a group of scientists and subject matter experts to study the California situation and make recommendations on the vaccination strategy, prioritizing premises for vaccination, and establishing a plan for how to use vaccine effectively should we not receive the amount we requested. We began our planning with developing standard operating procedures, focusing on 3 areas: vaccine handling, which includes receiving, storing, distribution, and chain of custody; vaccine cold chain, so how do we ensure the cold chain all the way out to the farm premises; and documentation. We asked ourselves several questions. What will be required in the form of animal ID? What other documentation might be needed? How long will the documentation be necessary? Depending on the vaccination strategy, we anticipate it may be necessary to track vaccinates throughout their useful life. We also asked how might we get real-time updates of vaccinated premises to our incident command so that they can be kept informed. Other considerations include vaccine security. So we anticipate needing to coordinate with our state emergency management agency to get security teams to escort vaccine shipments to the state warehouse. We also anticipate needing security at the vaccine distribution site itself. So we plan to use a third-party logistics provider for our state vaccine warehouse. We plan to station at least one state logistics person in the warehouse to oversee operations and be the liaison between the warehouse and the incident command post. In 2011, we exercised our ability to distribute vaccine during a simulated FMD outbreak. We had state personnel in a cold storage warehouse repacking vaccine for individual premises. What we found is that government teams are not very good at this. The more effective way is to contract with a third-party provider that distributes vaccines as part of their normal business operations. They are already set up for the cold chain, inventory, and shipping, and they are way more effective at this process and will increase the speed and accuracy with which we can store and distribute vaccine. In California, we use the Incident Command System to manage outbreaks of foreign animal disease. The Incident Command System helps provide an ad hoc organization needed to manage the outbreak and helps to reduce duplication of efforts and standardize processes. We have a state incident management team that includes blended resources from CDFA and USDA Veterinary Services. USDA also has national incident management teams that can deploy for a foreign animal disease outbreak. In California, we manage outbreaks in unified command with USDA counterparts. So it's helpful that we use the same incident command system training and organization so that we can blend together quickly when needed. We recognize the need to support different distribution strategies for vaccine. In this slide, it depicts several different organizational options, which include government vaccination teams that are made up of on-farm personnel that can be led by private practitioners or other temporary employees. In reality, we believe it will take a combination of these resources and many others to get this enormous job done in a short period of time. Our concept also includes an on-site validator. This would be a person from our incident command post who is assigned to each premises to oversee the vaccination and report back the status to the incident command post. In this way, all vaccinations would be overseen by a government official. Since vaccination is just one of many other disease outbreak critical activities that will be occurring simultaneously, we anticipate that we will need to engage other state and federal agencies to supplement our personnel resources. So what are our next steps? We need to update our standard operating procedures. We will be educating and training producers and responders on our plan. We plan to conduct an exercise to test individuals' capabilities to execute what they learned through training. And we need to test our coordination with the laboratory for broad EuFMD detection and surveillance. We want to look at capacity reporting and data monitoring. Thank you very much for your attention to this presentation. Please remember to leave questions in the chat, and Dr. Hollinger and I will do our best to get answers for you.
Fantastic. Thank you to Pam and Lisa for providing that presentation. And please do leave them questions for them to respond on the forum. Our final recorded presentation for today is another joint presentation from Beatriz Sanz Bernardo from the Pirbright Institute and Marlon Grant from EuFMD, and they will be presenting on faster vaccination, assessing a level of preparedness. Thank you.
Hi, my name is Marlon Grant. I work for the EuFMD. This work I'm going to present has been carried out together with Beatriz Sanz Bernardo at the Pirbright Institute, and it involves scoping work to assess the level of preparedness for emergency vaccination in EuFMD member nations. Vaccination for FMD is of course not a new issue, and work is carried out on regular intervals at EuFMD. They organize workshops, surveys, etc. And a new guide to the development of emergency vaccination plans is soon to be published. What's new here is that it includes other diseases than FMD. And I believe that increasingly the pressure to be prepared for emergency vaccination is increasing with the demand for more sustainable disease control options. This work relates to other activities at the EuFMD. In particular, work in the same component 1.3, which all deals with emergency vaccination, and all aim to increase preparedness for the use of emergency vaccination for fast diseases. The other activities in this component mainly relates to vaccine availability, which I'm not going to go into in this presentation. So this is about the scoping work done to understand the level of preparedness for emergency vaccination for fast diseases in EuFMD member nations, and to gather information on the barriers and constraints for including vaccination in disease response plans. The scope of the work is category 1 fast diseases, so that is foot and mouth disease, lumpy skin disease, peste de petit ruminant, and sheep and goat pox. And here you see examples of outbreaks of these diseases in member nations in the last 10 years. And vaccination is used for all 4 diseases in Turkey, and it was also used very successfully to eradicate lumpy skin disease from Europe in 2016-2017. What has been done so far is an online questionnaire that was circulated with EuFMD member nations' focal points in the summer of this year, and the next step is to carry out follow-up interviews. What I'm presenting today should really be seen as a preliminary result, as only part of the work has been completed, and ideally we would like to include views from more member states to be represented in this work. On this map, you can see in dark green countries that was asked to participate in the survey but didn't respond, and in light green the countries that responded. And links to the online questionnaire was sent to 42 countries and 15 returned full responses on this. And now follow some examples of questions and responses on the online survey. In this first example, you can see that all responding countries has a contingency plan for foot and mouth disease, but only few has got a contingency plan for PPR or sheep and goat pox. Next one is that the majority of countries state that accessibility to independently peer-reviewed information on the quality and key attributes of vaccines against fast diseases would influence the readiness to apply emergency vaccination. And as I mentioned, this work is ongoing at the EuFMD. Next example, we thought this result was interesting as only a minority of countries has estimated the rate of delivery of vaccination, which could imply that they have not calculated resources needed for emergency vaccination for different scenarios. And this is another area where EuFMD could potentially offer support. Now follows 3 slides on preliminary key findings. First, on the contingency plans. The results show that specific contingency plans for PPR and sheep and goat pox are uncommon. Also, emergency vaccination for FMD and lumpy skin disease is considered by 80% of responding countries, and many have already a defined strategy. This is not the case for PPR and sheep and goat pox, where only a few of the countries consider vaccination for these diseases. Regarding vaccine availability, most of the countries will rely on the EU bank with no other arrangements to source vaccines. We noted that national vaccine banks are not so common in this group of countries, and most have not approached commercial companies to source vaccines. Not all countries have a system in place to perform a rapid evaluation and approval of vaccines for emergency use. Also, as mentioned, countries are positive to having access to key information about available vaccines. And the next point is that we asked some questions about timing of procurement and implementation of emergency vaccination. And many would start procurement when there's an outbreak in their own country, or they state that initiation of procurement is not decided in the plan. Only few would start planning to procure vaccines in a scenario when there is an outbreak in a neighboring country. Another key finding we thought is that 73% of countries state that they would consider regional banks beneficial. Regarding vaccination strategies, the results imply that some countries would vaccinate all species with 100% coverage for FMD, which could imply that the scenario where doses are limited has not been considered in their plans. This is an area where EuFMD could support countries with priorities in different scenarios, where there is not possible to vaccinate all susceptible species. This could, for example, be in a multi-country outbreak. Also, a relatively high number of member nations would choose a vaccinate to cull strategy over a vaccinate to live strategy for FMD. Half of the responding countries has an exit plan for FMD, but only a few has an exit plan for other fast diseases. And the level of exit planning is something that we would like to investigate further in the follow-up interviews. Next slide is tactical and operational preparedness. The first point regarding estimated rate of delivery has already been touched upon. Also, the results imply that veterinarians would be used to carry out vaccinations. We would like to also investigate other options, as vets are likely to be a limited resource during an outbreak. Simulation exercises for emergency vaccination is uncommon, which could imply that the responding countries has not got as far as that in the planning process. So where do we go from here? So we are going to produce a written report with the key findings from the questionnaire to circulate with member nations. And then based on the results from the questionnaire, we want to organize group interviews with risk managers or contingency planners in member nations, and that's to be done early 2021. And the aim is to validate the results from the questionnaire and prioritize the gaps identified. It's also an opportunity to share knowledge and a good example amongst the representatives. We want to capture more detailed information about the gaps and needs in this area. And then we want to see if there are FAO or EuFMD tools already available that could support member nations in the preparedness work for emergency vaccination. And we also want to see if there's a need for projects or studies that could answer some of the questions raised in the scoping work. Something we have thought of already is how the EU Vaccine Bank could support a multi-country outbreak. So what prioritizations are needed to make the best use of the doses available, really. Okay, that's it from me. Thank you all very much for listening. And please write any questions that you may have in the online chat. And then we hope that some of you will be happy to contribute to the work going forward with the group interviews. And Beatrice and I would like to thank the colleagues at the EuFMD for their contributions so far, in particular Pam Hallinger and David Mackay. And I wish you all a good rest of this open session. Thank you.
Thank you very much for an interesting presentation, Malin. It's good to see that you are in touch with what is happening in the EU member states. Now we will get ready for the round table. And before that, I will thank all of the presenters, and you will see them back into in the screen in the round table. We've had some excellent presentations today. In case the audience has some questions, please put them in the forum so that they can be asked during the round table. And with that, we will be back in just 1 minute. Welcome back, everyone. Now we are getting ready to the round table. And for the audience, please use the chat box of the website to post your questions so that we can get them in this forum and we can discuss about things. I was thinking I would start with a question to you, Naju. You discussed about the early immunology of vaccination against foot and mouth disease in pigs. Where can you share where you see the next phases of the expansion of this work in terms of booster vaccination strategy or ultimately downstream protection from challenge?
Thanks, Sten, for the question. Can you hear me? Yep.
Yes.
So we have plans to actually do this, extend the study for the other downstream days, maybe day 5, day 10, day 14, to see how the gene upregulation or downregulation happens, and then how the BTM modules move up and down to effectively see the difference between the intramuscular and the intradermal vaccinations. We also want to differentiate between a single vaccination versus the prime and boost, because in pigs we need 2 vaccinations. That's very essential for long-term immunity as well. And also, if we find any interesting gene modules or something, we would like to validate that in a protective response test as well, like a vaccine efficacy test, with comparing the intramuscular and intradermal as well. So we have plans in the pipeline to further extend these studies.
Thank you. And that's going to be very interesting to follow in the next coming years. And Emma, I have also a question for you. For Sarah. Can you elaborate on what were some of the challenges with accessing data to parameterize the model? For example, are there some specific areas where improved data would be of greatest benefit to enhancing model predictions?
Hi, thank you for the question. Yeah, so that was one of the biggest challenges of the model, is getting sort of accurate data, particularly the actual FMD outbreak numbers, but also just consensus data on populations and like livestock sector splits. So, like knowing all the different dairy sectors and the beef sector and pastoralists and smallholders, because they all have different FMD instances. And additionally, sort of knowing like livestock density per region, as this will have a big sort of effect on the foot and mouth. So yeah, there's lots of challenges facing the data, and we're looking into trying to sort of improve that as we are developing the model.
Okay, yeah, that's good. And another question, what different target groups do you envisage would make use of the predictions from the model? Veterinary services, perhaps, the main target group, but could also be used by the private sector or vaccine producers. What do you think?
Yeah, so yeah, another good question. I think it can be used, but hopefully it'll be able to be used by everybody involved, who's got a stake in foot and mouth control. Yeah, definitely the veterinary sector, but also manufacturers, if they've got sort of like a ballpark. It's an estimate number. It's not intended to be accurate, but it's there to say, look, this country would need this amount of money, this amount of vaccine. And likewise for government and policy, it would be a good way to sort of benchmark and say, last year you used this, but actually we sort of recommend that you use this much vaccine, and it will help you in your control. There's a big supply-demand mismatch with vaccine, and I think it would be a good way to sort of highlight that and sort of provide information for donors, policy and vaccine producers.
Okay, thank you very much.
Thank you.
Anybody else at the round table with a question for Naju or for Sarah? Okay. Yes, Sam, you've got a question. Thank you. Come on.
I have a question for Nagendra. Nagendra, to what extent do you think your research will have implications for vaccine platforms for other diseases?
Thanks, Sam. It's very important to see that pigs are seen as major food source in Southeast Asia, Asia, and other countries. There are some intradermal vaccines already in the market, but the thing is, FMD is the important disease in Southeast Asia. And the conventional vaccine that is delivered intramuscular, as shown and from the published literature, creates a lot of issues with vaccination, with nodules and other things. So the farmers have refused vaccinating these animals, and this has impacted the control programs largely. Another thing is if we can combine different vaccines as an intradermal formulation, that means we don't have to visit the farms many times to administer different types of vaccines, but we can put in formulations where different antigens are mixed together in a deliverable format with a better adjuvant, and then we can target those immune responses that are not normally triggered by the conventional vaccine, especially the cell-mediated immune response and the long-term memory response. So that would be the aim to achieve, to have long-term immune response. And then if this works, we can take it to another species, maybe cattle, to deliver the vaccine intradermal and produce a long-term response. So it makes life easy for the vaccinator, no needle stick injuries, no need of cold chain if that is the way to achieve it. So there are certain pathways that we can take this research through.
Well, thank you. Thank you for this reply and a good question. Any more questions from the group here?
I can jump in here. I have a question for most of you, but I'm going to be cheeky and I'm going to ask Sam a question to start with. He's actually my boss, so I'm being bold.
Yes,
so Sam, are there any lessons from the COVID-19 pandemic to guide our contingency planning and investment in livestock vaccine banks? And I guess how can the epi modeling simulations take these into account, these lessons into account?
Thanks, Chris. It's such a big question and a good one. Really appreciate it. Look, I've been thinking about this a lot, and COVID-19 has affected all of our lives in different ways. And I really answer this question as a citizen, not as a bureaucrat. And I think the main issue around public policy here is around transparency and good communication. So all of us have faced decisions being made that have affected our lives, that have been made by public servants and politicians under a lot of pressure in whatever country we are, and facing a significant amount of uncertainty. I think we can do a lot better in the animal health sector about talking about what we know, about what we don't know, and the basis for the decisions, and really empowering people. I guess my 15 years in the public sector, a lot of our planning has been around how government and private industry would make decisions for farmers on the ground. And I'd really like in some way for us to start to hear their voices and involve them in that decision making more. So I think when we talk about vaccine banks and distributions of vaccines, all of which are questions that we'll learn a lot from in 2021 with the response to SARS-CoV-2, I think we really need to start to engage people and really expect more from the average farmer, and expect more from the average member of the public too.
Thanks, Sam. That was a really genuine answer. That's really nice to hear. Sten, would you like to jump back in?
Yeah, well, I would like to follow up a little bit on the COVID situation, because, well, as we saw from Australia, you base your contingency plans and your surveillance plans on epi models and risk assessments of known diseases. But what about the unpredictable, as we've seen with COVID-19? What then becomes important for governments to consider? What are the real values of good contingency in that situation? And that's the question for everyone.
Well, I might have a first go at that, Sten. I suppose I have maybe a slightly unconventional approach to contingency planning. I think it's a very useful exercise to undertake. It kind of gets us to think, about what the processes we need to follow. And from my way of thinking about this, it's really agnostic as to what the particular challenge it is we face. Whatever the disease situation or problem, or even in a laboratory like ours where we cut across from animal health to food safety, many of those kind of incidents have the same kind of characteristics, and I kind of think of these as three phases. There's the immediate thing of, you suddenly realize you have an incident, you have an outbreak, you have an event. There are communication challenges around that. Then you very quickly have to move into, how do we get to the bottom of this? And again, I think a lot of these things are the same, the same kind of challenges you're going to face. Who are the people I need to communicate? How do I mobilize teams? And last but not least, and particularly for countries that export a lot of food or livestock, how do we show to the international community that this event or incident has passed? How do we show that with confidence? So I think it's useful to do this, but the great, I suppose, the military strategists say that battle plans never survive first contact with the enemy. I think we all must have to be prepared to put the plan in the bin. It's the thought process, how we actually elaborate it, that's more important.
I think that's very true. And well, anybody else which will have a go on this question? Yes, Naju?
Yeah, I have a question for Donald. With every country preparing its own test materials or new kits or new developments, how do you take a call on how to verify in a short notice during a pandemic which one to use, who will be using these kits, and then do we leave these kits to be handled by a private operator or a private testing lab? So how do we control these developments, especially in developing nations or third world countries? It's going to be a big ask there.
Yeah, it certainly is. I mean, one of the challenges facing everybody at the moment is the multiplicity of these test kits. And while some of them may come from reputable suppliers, there are also, I suppose, less scrupulous operators who see the opportunity, for market advantage, and people, there's kind of a huge thing to get product out onto market maybe before it might be fully verified to avail of first mover advantage. So, I mean, it is a challenge. As to who might use these, I don't know that we have any way of really controlling who's going to have access to these. Certainly at the moment, and from what I know of in Ireland, different people are using rapid tests in different settings for different purpose. And it is a matter of concern. And I suppose particularly if these tests are not being used correctly, that maybe they're giving people a false sense of security. On the last point you mentioned, and I mean, I would say it's not just in developing or emerging countries that this would be a challenge. I think it's a challenge for everyone. But I think there's a very strong case to be made for pooling our collective resources here. We've seen at national level, well, particularly across our acute hospitals, that by people actually communicating with each other, sharing information, they were much more quickly able to validate systems. And, we could, for example, and we have organized these kind of exercises requires coordination.
Okay, I think we had a small issue here. The last part of your reply was frozen away, Donald. Maybe you can
Okay.
repeat.
Okay. Well, I suppose the critical thing in that, if you want to attempt any form of collaboration across different entities, is that you have some coordinating mechanism. So I guess we need something at international level, you know, and I suppose an organization like EuFMD is ideally placed to play that kind of networking role.
Very interesting observation, and I do agree with you of the crucial role that EuFMD can play. There's another question here for you, Donald, from Ronan O'Neill. PCR is a very flexible technique capable of dealing with almost all biological threats. As part of national contingency planning, should efforts be made to strategically ensure very large PCR capacity always exists, and in either human and animal health to allow timely switching to a pandemic role if needed?
Well, I would answer yes to that, Sten, but I suppose what are the practical impediments to trying to get there? And I suppose speaking from this country and my experience of working with different people, well, we have no fewer than 27 different testing laboratories involved in providing PCR testing at present. I suppose if you were to move to that, and it makes eminent sense that you would pool this capability, and it could be applied on a cross-sectoral basis. But who is going to host it, and who is going to stop doing it, and how are you going to arrive at, at an answer to that? You have other, I suppose, related issues like sharing of data that comes up with this. I mean, it makes absolute sense. I mean, it's something that we have looked at in terms of our reference laboratory capabilities. And probably the area or space where we have had most experience of trying to create that kind of thinking is in respect of near real-time sharing of whole genome sequence data with our public health counterparts, particularly in terms of foodborne enteric pathogens. But it does take, I think you have to start small. I think you have to take some examples. I think you have to build up trust. But I think for a small country like Ireland, it would make huge sense that we pooled our collective resources. It would give us redundancy and resilience in those testing systems that we simply don't have, trying to maintain a multiplicity of different centres.
Okay, yeah. That's so it's a stepwise approach to become more resilient. And there's another question here from Dr. Fabrizio Rosso, who thanks you for a very interesting presentation. Is a shared diagnostic bank a possible solution for responding to a multi-country disease spread and the need for rapid scale-up? What do you think about that?
Thanks, Fabrizio. Well, I think you might still have some of the same issues as you're likely to have if you're stockpiling reagents at national or laboratory level. You have the shelf life issue with these products, and then you have the issue of what exactly are you going to stockpile here? It would make a certain amount of sense perhaps for generic materials that have wide scale application, you might say, but then on the other hand, thinking about it, what about those unusual minority diseases that maybe we don't ordinarily keep material for? Maybe that's what might be kept in an international bank. Yeah, I don't know. I don't think there's any easy answer to this, and I think you're left with the same challenges at international as at national or laboratory level.
Thank you for this reply. And then for Malin Grant, a question. How do you see the role of the EuFMD in preparing better for vaccination in each of the EU member states?
Thank you for that question, Sten. Yeah, I mean, it's really the main objective for the ongoing work is to find out what are the needs in the member states and what do they need from EuFMD. But I think already the EuFMD networks is really an opportunity to interact and share good practices between countries in a nice, friendly atmosphere usually. And then there's also the training opportunities that the EuFMD offer, the tools. There's both the existing ones and ones that can hopefully be developed based on the needs that we see in this project. We got the discussion of allocation of vaccine doses. I think EuFMD has a role to play there, for example, in a multi-country outbreaks. How do we use the EU bank doses in the most effective way? There's ongoing discussions that maybe the disease spread model for FMD, the EuFMDs, could be helpful here, and lots of other aspects. I think the work on vaccine availability is also another large part. So I think the list could be made quite long.
Yes, it certainly can, and as I see it, EuFMD can also help sharing expertise, because when it comes to these diseases, there's not much experience in any of the EuFMD member states. Not very many of the old guys left that took part in foot and mouth disease many years ago, and some new diseases creeping up in Europe where we do not have a lot of knowledge. So what's your comment to that?
No, I think that's a really good point, and I think there's something that we would like to maybe bring into this work. Now you mention it, because a lot of our focus has been, okay, how are we prepared? But then in an actual, when you're faced with an outbreak and you need the expertise, then how can you prepare to what are the channels to interact with the EuFMD, and how can EuFMD actually support in the event of an outbreak? I think that's something that's really good to discuss.
Yes, thank you. Anybody else has a question?
Can I bring us back to Sarah? I've got a quick question for you. So how would you model demand for vaccine in free settings? Would you need any other models, or could this approach be used, the approach that you presented, be used for this purpose in addition to endemic settings as well?
Hi, thank you, Carissa. Yeah, I think so. The model is based on PCP, but PCP is a proxy for sort of your control strategy. So in theory, you could, if you got all the accurate details of the consensus data and everything like that, you could just plug it into the model and say, oh, so we want to vaccinate quite strongly, so then we'll go in hard and we'll go for PCP4, which will have a certain vaccine coverage prophylactically and emergency wise. So yeah, there is some flexibility in how you use the model. It's not just, it's very versatile. It can be used for lots of different countries and everything.
Fantastic. And I think I saw Nagendra. Did you have your hand up before to ask a question?
Yes, thanks, Carissa. Question is for Sam. How does the government of Australia interact with the private sector in contingency planning for emergency animal diseases? You have an interaction with the farmer level, but there are different supply chains before the product reaches the customer. So how does the government interact with the private operators?
So, as I mentioned, we've got relatively good connections with our peak industry bodies. But Nagendra, I think you've really asked quite an insightful question within Australia, because there's various actors within supply chains that don't represent farmers that have more and more power when it comes to decision making. So I'm thinking about our major supermarkets, for example, to some extent major processing plants as well. And I think there's more work that we can do to try and engage these decision makers. Certainly, there's some engagement, particularly around the supermarkets. But yeah, there's a lot more that we can do because, of course, whatever the public messaging that we might use in a disease outbreak, it's really going to depend on the trusted voices that the Jamie Olivers of the world, who has represented one of our major supermarkets in Australia, that will really be heard, probably much more than the Australian CVO, much more than key decision makers and bureaucrats. So it's a work in progress.
Thanks, Sam.
Thank you very much. Very interesting. I also think that working with the private sector becomes more and more important. As well, I'm a bureaucrat as you, Sam, so it's also very close to me, the subject. And then one question for Donald again. You're a very popular guy in this round table. Did you have to make tough prioritizations between testing of SARS-CoV-2 compared to other animal diseases during this crisis? And if you didn't, have you thought about what you would do, how you would prioritize?
Yeah, well, I suppose the timeline for us, we started handling samples and doing this data entry step from about the middle of March. We discontinued doing all COVID-19 related work within about 3 months because there was testing capacity elsewhere. And we attempted to establish a pipeline for COVID testing, but we couldn't get the reagents for our automated extraction system that we're most comfortable with using. We were provided by public health authorities with an imported product which was much more labor intensive and much lower throughput. And it was so clunky, in fact, we thought that we could use our resource better elsewhere. But at the same time, we had precious little of these reagents to deal with, as I mentioned, an avian influenza outbreak. So, I mean, we did have some hard choices to make over that period of time. Around how we would use and ration out the small amount of resource we had. But I suppose, like everybody at that time, this was #1 national priority, trying to address this major human health issue and, develop testing capability. That was the immediate and most pressing challenge for government in March and April of this year.
Okay, thank you. And then there is a question from the audience, from YouTube, from Evelyn Hunje. It says possibility to make vaccine by pool. Anyone knows what the question is? No. So, well, we don't understand the question. So sorry, Evelyn. Well, and then we have a question for Naju from Dr. Aldo Decker. Why do you propose to boost? In the Netherlands, we never used boost vaccination. We vaccinated with 10 to 20 PD50 per dose vaccine. The response was sufficient for 1 year protection.
Yeah, we find this question regarding the experiences of Europe with the experiences in Southeast Asia and Asia. We should understand that the vaccines are not the same. The quality of vaccines that were used in Europe and the standards that are in Europe are not the same as the standards that are in the third world countries. Another thing is there the condition of the animals are not great. The nutritional status is not good. And these animals require at least 2 vaccinations to build up their immune response. That's the normal thing that is in the field conditions in these areas.
Yeah.
So we cannot relate directly the experience of Europe with the experience that we have in Asia. So that's one reason. That's why the booster dose is very important. Given the quality of the vaccine, not all countries can afford a more than 6 PD50 vaccine. The normal vaccine used there is around 3 to 6 PD50, which is a normal vaccine there. And these vaccines that are used in these countries are mostly donor supplied vaccines. So they come with a certain caveat there as well. So it's a different experience that we need to, and in our test results, we may find that prime alone works very well, prime boost is not required. But to reach that point, we need to compare like with like, especially with the intradermal vaccine, which is a new type of vaccine. So it's part of the research experience. And then once we find a tangible evidence that only prime is enough, and with the quality of the vaccine being good, then we may recommend a prime vaccination. So that could be my response.
Okay, thank you. That's a very good point you're making. Thank you for this, and thank you to all the presenters for being with us this morning and sharing and having an open and discussion and being transparent. We really enjoy that. And also thank you to Carissa Miller, who took us through this morning as the moderator. And Carissa, you will be stepping down from now on, and in the afternoon we will see Buddha Amadi, who was working behind the scenes this morning, but he will be on the screen this afternoon together with me. And now we will have a 2-hour break. We will be back at 3 o'clock Central European Time. And I will wish you a nice break, all of you, and go and refresh yourselves and have a good time. And if you have extra time, go and check out the website and the virtual space and explore the contents of the EuFMD platform. Thank you.
Thanks very much.
and good afternoon. Welcome back to our afternoon session of the third day of EuFMD's Open Session 20, entitled Livelihoods at Risk in a Faster World. My name is Buda Ahmadi. I am a veterinarian and animal health economist, and I have the pleasure of being the moderator of this session this afternoon. We had a very interesting session this morning with the theme of vaccine security and critical resources for emergency management. We will continue this afternoon under the same theme. We will have a number of very interesting presentations for you, which we hope you enjoyed. And at the end of it, we will have a roundtable discussion, which lasts about 30 minutes or so. Before introducing the chair of this session, I would like to ask you, if you have not done so yet, go to our website and register in our discussion forum, which you can post your questions to our presenters this afternoon. Please ask your question there, and we will gather the questions and we'll direct them to the presenters of this session. So without wasting time, I would like to introduce the chair of this session, Dr. Sten Mortensen. Sten is a member of the EuFMD's Standing Technical Committee, and also he is the head of Animal Disease and Control at Danish Veterinary and Food Administration. Sten, please, floor is yours.
Thank you very much, Buda. Good morning, everyone. Good afternoon, good evening. Welcome, everyone, and welcome back. This session was planned by Pam Hollinger from the EuFMD. As she's based in California, we hope that she'll be joining later this afternoon as the sun gets up in California. But we will go on with the program, and I hope you will enjoy the afternoon. And I would like to introduce the first speaker, Dr. Mike Francis from Biovac Consulting, based in the UK, with a presentation from bench to field, rapid veterinary vaccine development and platform technologies. Over to you, Michael.
Thank you. Thank you for the invitation to give this keynote presentation. My name is Mike Francis, and what I'd like to do today is to talk about the veterinary vaccine development process, how we can potentially accelerate that process, and why platform technologies are so important to the production of new generation vaccines in the future. If I can have my first slide, please. So what are the key considerations that we need to take into account when developing a new vaccine? Well, first of all, we need to identify that unmet market need for the new vaccine, and in doing so, to develop an outline product concept. When we develop an outline product concept, we should involve cross-discipline input from research and development, from marketing, from manufacturing, and from finance. We can then produce a clear target product profile and associated timelines for the project. And the product should always be designed with the end user in mind, and that involves customer input throughout. Next slide, please. So the vaccine development process itself can be split into two stages. Within the research stage, which can take up to 5 years or more to complete, we have a discovery phase and a feasibility or pre-development phase. And within the development stage, which can take 3 to 6 years in Europe, we have an early phase development, late phase development, and a registration phase. Next slide, please. The veterinary vaccine discovery phase involves developing an understanding of the pathogen and the immune response within the intended target species. This in turn will help in the development of a suitable challenge model designed to mimic natural infection that can be used to assess the protective efficacy of an experimental vaccine. Once an appropriate immunogenic formulation has been identified, then the project can move forward into a feasibility phase. This involves proof of concept, safety and efficacy studies within the target species for the vaccine. And it's important to ensure that the immune response elicited by the vaccine correlates with protection within the host, and thus an optimum immunization strategy can be developed. This will hopefully result in the identification of a lead vaccine formulation. Next slide, please. As soon as there is confidence that the experimental veterinary vaccine candidate elicits an immune response that provides suitable protection, then the formulation can be moved into early phase development. It is necessary at this stage to prepare and safely store a pre-master seed and a master seed of the lead antigen. The early phase development is focused on addressing 3 key elements of a regulatory dossier, which within the UK and the European Union are broadly defined as quality or manufacture, safety, and efficacy. The quality data package should address purity and consistency of the final manufactured product and ensure that the batch of vaccine is fit for purpose. The safety testing data will be dependent on the nature of the vaccine, for example, whether it's inactivated, live attenuated, or recombinant. This will generally include single, repeat, and possibly overdose studies carried out with maximum potency product in minimum aged animals. Further studies will also be required for animals in pregnancy or birds in lay. Finally, the efficacy data should include onset and duration of immunity studies conducted with minimum potency product in minimum aged animals. It may also involve efficacy in the face of maternally derived antibodies, a study of the requirement for a booster dose, and an investigation into the immune mechanisms involved in the host's response to the vaccine. Next slide, please. During the early phase development, the pilot scale manufacturing process should be validated and scaled up to levels more reflective of the final manufactured product. This should eventually result in 3 GMP manufactured consistency batches and finished filled product that can be used in stability tests and field trials. Next slide, please. Data from the early phase development is used to apply for permission to conduct field trials as part of the late phase development and to ensure that the vaccine is both safe and efficacious under field conditions, which are designed to mimic its everyday use. The details of the trials will depend on the species for which the vaccine is being developed. The registration phase will be dependent on the regulatory strategy and the intended markets for the product. And it's important to consider the recommended dose and route of administration for each species and category of animal in which the vaccine is intended for use. Next slide, please. The maps that I've shown you today have principally focused on the development of a vaccine for the UK and European Union markets, and they will lead to the generation of a product data package that would be used to compile a regulatory dossier for marketing approval. Other global markets will have their own regulatory agencies and specific regulatory requirements. These maps can be found at www.vaccinedevelopment.org.uk, and the details of the process that I've talked about have been published recently in the journal Vaccine. Next slide, please. So what I'd like to do is just to share with you some potential ways in which the process itself can be accelerated. If we start with research and development, it's important to have an experienced project manager and to develop a comprehensive project plan. This project plan can be used to identify the critical path for the vaccine development and to look at ways in which that critical path can be shortened. You should conduct early proof of concept studies in target species, and you should establish early correlates with protection. It's important to use pilot scale vaccine produced according to the final manufacturing process within the critical trials so that they can be used within a regulatory submission. And you can often run safety and efficacy trials in parallel in order to shorten the timelines. Next slide, please. If we consider the manufacture, wherever possible, you should utilize existing technologies that will be familiar to the regulators and it's very important that you record all reagents used throughout the vaccine development process. You should prepare a master seed early on prior to any animal trials, and again, this will help you to use the data for regulatory submission. And you need to define the outline manufacturing process at an earlier stage as possible. You should also establish a potency test and begin stability studies as soon as possible, because these can often be rate limiting in any regulatory submission. And if possible, you should also scale up the process in parallel with the safety and efficacy studies running in the field. That is to say, you do it at risk. Next slide, please. Regarding the regulatory process, it's very important that you talk to the regulatory agencies at an early stage. And there are various opportunities to do that. There's an innovation task force briefing within Europe, which can be used to discuss new vaccine technologies. There are scientific advice meetings and pre-submission meetings. You should generate sufficient target species safety data and provisional efficacy data at an early stage so that you can gain field trial approval. And within Europe, you should consider using the minor use, minor species regulatory pathway, because this can shorten the timelines to product registration. You should also, where possible, use an established and well-characterized vaccine platform that will be familiar to the regulators. There is a new regulation coming into force in January 2022 in Europe, and this has one of the aims of this is to support the vaccine innovation and platform technologies in the future. Next slide, please. So if we go on to explore different vaccine technologies, both conventional and novel platforms, well, of course, conventional vaccine technologies broadly fall between inactivated killed vaccines and live attenuated vaccines. Novel platforms such as subunit viral proteins and virus-like particles can mimic the inactivated virus vaccines, and viral vectors and nucleic acids such as messenger RNA, self-amplifying RNA, and DNA can be used to mimic the attenuated virus vaccines. Next slide, please. What I'd like to do now is just to give you one example of a One Health approach to Rift Valley fever vaccination using a live vector vaccine approach. This is work conducted by George Wurimwe, who is a principal investigator at the Jenner Institute, and it's in collaboration with the Pirbright Institute. Rift Valley fever is a Bunya virus, and it's a mosquito-borne disease. It's also a zoonotic infection, and it infects livestock, including sheep, goats, cattle, and camels, and also humans. The disease occurs in Africa and the Arabian Peninsula, and it causes a febrile illness progressing to a meningoencephalitis, a retinitis, fulminant hepatitis, blindness, and hemorrhagic syndrome. It can cause 20 to 50% mortality, and it's a World Health Organization notifiable disease. There's currently no licensed human vaccine for Rift Valley fever, but there is an attenuated Smithburn vaccine, but this does have problems with its safety in pregnant animals. Next slide, please. So the approach here, as I said, was to use a viral vector, and the viral vector here is a chimpanzee adenovirus called Chatbox1, and this particular vector is a non-replicating vector due to E1 and E3 gene deletions. Inserted into the vector are the GN and GC genes responsible for immunity to Rift Valley fever, and these are encoded within the viral genome, and they do not form part of the structural virion. It's interesting to note that this particular vector is being used for a number of other human vaccines, and of course it is one of the lead candidate vaccines for COVID-19. Next slide, please. So the concept here was, can we produce one vaccine for multiple species? That is to say, a single vaccine that would be used for livestock and for humans. Next slide, please. And the advantage of this vector is that when it goes into the host and enters the cell, it expresses the foreign proteins, and these proteins can be detected by antigen-presenting cells and presented to B cells to produce antibodies. And the proteins can also be degraded, combined with class 1 molecules on the surface of the cells and presented to CD8 positive T cells to elicit a cytotoxic T cell response. So this particular vaccine will elicit both antibody and cell-mediated immunity. Next slide, please. If we look at some of the antibody data in cattle, sheep, and goats, and this is data obtained at 28 days after a single vaccination, we can see here the virus neutralizing titers are high within all 3 species, and we've got equivalence here between the Chatbox vectored vaccine, the Chatbox vectored vaccine with matrix Q adjuvant, and the positive controlled Smithburn vaccine. They're all very similar, and there's no obvious advantage in adding an adjuvant to the vector. If we look at the placebo, of course, there's no response in the placebo vaccinated animals. Next slide, please. If we then go on to look at viremia after challenge in vaccinated pregnant animals, and here we're looking at pregnant sheep and pregnant goats, you can see that the mock vaccinated animals have a clear viremia that peaks around 3 days after challenge, whereas those vaccinated with a Chatbox vector have no viremia detectable at all up to 14 days post-challenge. So the vaccine clearly protects against infection, protects against viremia, and it also protected these animals against any abortion caused by the Rift Valley fever challenge. And the vaccine was also shown to be entirely safe within these pregnant animals. Next slide, please. Another important aspect of a vaccine, of course, is its stability. And this is some recent work from an International Veterinary Vaccinology Network pump priming grant that we received. And it showed us that based upon in vitro data, vaccine potency of a liquid formulation is shown to be stable for at least 30 weeks, 30 degrees centigrade for 1 week, and for a month at 20 degrees centigrade. Additional in vivo data shows no loss of immunogenicity of the liquid formulated vaccine after that month storage at 20 degrees centigrade. Furthermore, if you go on to lyophilize that vaccine in a stabilizing formulation, it's stable for at least 6 months at 30 degrees centigrade. So we've identified 2 options here for delivering this vector vaccine outside of the cold chain. Next slide, please. Next slide. So the benefits of the vectored Chatbox vaccine are that it's a single shot vaccine. It can be used in multiple species. It produces a good antibody response and protective immunity. And it can also prevent viremia within the host and abortion within pregnant animals. It's also been shown to be safe in pregnant animals, and field trials are currently ongoing in Africa to test its safety and efficacy in larger numbers of animals. The vaccine has good stability, and it's interesting that the same master seed can be used to produce the human vaccine. So the vaccines will be identical, and the manufacturing process will be similar up to the bulk harvest stage, at which point the human vaccine will undergo further purification. But it's a true One Health approach to vaccination. Next slide, please. So novel vaccine technologies often find their first application within veterinary vaccines. Recombinant technologies such as subunits, virus-like particles, vectors, and DNA have all been licensed for use in veterinary medicine. Novel adjuvants, innovative delivery, and stabilization are also commonly used in veterinary vaccines. And there are veterinary regulatory guidelines to facilitate new technologies, including the use of live recombinant vector vaccines and DNA vaccines. Having said that, it's worth noting that the majority of new vaccines are still based on conventional inactivated and attenuated approaches. Next slide, please. So if we just think about some of the firsts that have occurred within animal health, some of the biotechnology firsts, well, back as far as 1965, production of foot and mouth vaccine using BHK suspension cells was really the forerunner of many biotechnology processes that were to follow. In 1982, the first recombinant vaccine was produced against E. coli in pigs, and in 1988, a recombinant subunit vaccine was produced against a retroviral disease, feline leukemia, in cats. Throughout the 1990s, a range of canary pox vector vaccines were licensed, for example, against rabies, feline leukemia, and equine influenza. And in 2005, the first ever DNA vaccine was licensed for use against a viral disease in salmon. In the same year, the first plant-produced vaccine was licensed against Newcastle disease in poultry. And more recently, in 2017, a 3-component viral vector vaccine has been licensed against Newcastle disease, infectious bursal disease, and Marek's disease in poultry. Next slide, please. So what should we take away from today's talk? Well, first of all, it's important to generate a clear target product profile for a new vaccine. And this is sometimes called an SPC, or summary of product characteristics. It's important to consider the market requirements for the new vaccine, and commercial input is essential throughout the vaccine's development process. You should have an early interaction with the regulators to facilitate the regulatory process and smooth the pathway to registration. And you should always investigate various options to accelerate the development of any vaccine. And in order to do this, you need to be adaptable and quick to respond to changes. We should note that significant innovation already exists within commercialized veterinary vaccines. And I feel that novel platform technologies will be essential for the rapid development of new vaccines against new emerging diseases in the future. Next slide, please. Thank you very much for your attention.
Thank you very much for a very interesting and clear presentation. I find that in the chat box we have a question from Mortensen, who thanks you for a very clear presentation. It is still unclear to him how the efficacy of vaccines is tested in the target species during the clinical trials. How is the control group defined? May that be different for veterinary and human vaccines in development?
Yes, I mean, there are a number of questions there. I think within clinical trials, certainly within Europe, we always aim to produce an efficacy and a safety data package when we're conducting those trials. And in terms of getting an efficacy package, one can either look at surrogate markers of efficacy within the vaccinated animals, or hopefully rely on some challenge within the field to show that there has been a protective efficacy, and that would be the ideal situation. Not always possible, but that would be the ideal situation. What was the other thing? Yes, I mean, I think the control group generally would be either a placebo control or preferably an existing marketed vaccine, which you're comparing your vaccine against, to look that it's got comparable immunogenicity and safety, and hopefully improved immunogenicity and safety within the host. And in fact, the field trials within veterinary medicine, because we've already done a lot of studies in target species, are a little bit more predictable than human trials, because when we go into the human trials with human vaccines, of course, we don't have any real clear idea of whether the markers we've got of immunity will provide protection within the humans. Whereas within veterinary medicine, we have usually previously done a live challenge study in the target species.
Mm-hmm. Yes, thank you very much, Michael Francis. And I'm sure there will be many more questions at the round table, but for now we will move on. There's a small technical problem with the next keynote presenter, so we will wait a little bit with that keynote, and I will give the mic to Buddha to introduce the next presentation. Thank you.
Thanks very much, Sten. As you mentioned, there's a technical issue, but my colleagues are working on it. So hopefully we'll have Dr. Pascal Houdelet on live very soon. But meanwhile, I would like to introduce Dr. Alistair King to present his recorded presentation. Dr. King is the Director of International Veterinary Health at Merck Animal Health. And the title of Dr. King's talk is Prioritization of Vaccines, Approaches to Maximizing Impact. I would like to give the floor to Dr. King.
Good morning, good afternoon, good evening. Thank you for joining my talk on prioritization of vaccines. And really what I want to do is think about how do we maximize the impact based on the resources that we have available. I haven't got any perfect answers because there are no perfect answers. Everyone has different situations, but I want to talk through some of the considerations we need to have. When we think about transboundary and emerging diseases, often the approaches we talk about is getting herd immunity up, and we want it over 80%, 70, 80% in order to try and eradicate disease. And this leads to an approach which really is vaccinate everything as quickly as possible against every strain that's available. In theory, this is fantastic. This is a great thing to strive for and an ideal, but we're faced with resource constraints in almost all situations. Those constraints are different. The amount of available quality vaccine that can really do the job, that is definitely a big constraint. It's not possible to produce as much vaccine as is required at the moment for everywhere. Then we've got the issues of matching the vaccine to the strains. So that's either because the circulating strains are different or because we're using heterologous or homologous vaccines. These kind of issues lead to choices that we are making. We're looking at funding. It costs money to run vaccination campaigns, not just buying the vaccine itself, but the manpower that's needed, the other equipment, the logistical efforts to get vaccine to where it's needed. All of this costs a lot of money. And pure manpower. Having enough people who are trained, who know how to give vaccines, who have the skills and the knowledge. All of these are massive constraints. So when we're faced with those constraints, how do we best maximize what impact we can have within the constraints that exist? We did some work a few years ago with the University of Wageningen in the Netherlands and Dr. Langay and Dr. Ron Bergelet. And this was really interesting. We did a modeling program looking at FMD, in this case, foot and mouth disease. But we based it on a susceptible, infected, recovered model. And we looked at how much it would cost over a period of time. And each quarter in this graph is a quarter of a year, 3 months. So this is looking over a long period of time, 5 years, to see what is the most cost-effective approach to getting disease under control. And we looked at no vaccination, obviously. We looked at using high coverage, basically trying to get everything vaccinated with a high potency vaccine or high quality vaccine. We looked at high coverage with a low potency vaccine. We looked then at low coverage, so doing a much smaller percentage of the animals available, but with a high potency vaccine, and then a low coverage with low potency. This paper has been published, and if anyone wants a copy, please do get in touch with me. You would think that the more cost effective, well, not cost effective, but the cheaper option would be to use a low potency vaccine, which is inherently cheaper to purchase. But actually what you see here is clearly using a high potency, high quality vaccine and high coverage is very cost effective. That's the thing that gets the disease under control the quickest, and over 5 years is going to cost you the least. But the next best option is actually still using a high potency vaccine, but using low coverage. That getting out there with something that's going to get the disease under control very well as a vaccine, but not doing everything, not getting your 90% of animals vaccinated, that is still more cost effective than using a low potency or low quality vaccine that doesn't do the job. And actually using those low potency vaccines can cost more over time than not vaccinating at all. So without prioritization, but making cuts and choices in other areas, actually you can end up costing more in our disease control and letting the disease spread rather than get under control. So this is why we need to think about prioritization. What's the best way to use the resources that we have available? Of course, the question is, what does prioritization mean? There are loads of different things. For different people, it's not the same. You can think about prioritization by country. When we talk in the global level, like we're talking now, EuFMD, we're looking at something that's international, then we have to look at which countries do we see the biggest risks, have the biggest problems. Those are the ones to prioritize. You can think about disease. Is the priority foot and mouth disease, or is it lumpy skin disease when that suddenly decides to jump borders and spread and that starts to cause problems? Is it African swine fever, which we don't even have good vaccines for at the moment? What do we need to do with diseases? Is it by strain? We look in Africa and foot and mouth disease. Do we need to worry about the South African territory strains when those aren't the ones that are causing the majority of the disease and the most serious disease? Maybe those are tougher to get under control, so maybe we should prioritize the ones that we can do something about. Is it by vaccine? Is it by the vaccine type? How is it made? What does it do? Is it by procurement? Procurement teams in countries look at cost. So you can put everything else together, but they're going to go for the cheapest option because they're trying to save money. But that is a prioritization. Or is it by your objectives? Do you want to eradicate the disease? Do you want to get it under control? Do you want to stay free from the disease? All of these questions come to what prioritization means for you as an individual. But it's important to think about these things and be aware of them and know what you're trying to achieve. So how do we then plan our prioritization? Surveillance. That is really important. Where is the disease? And we still don't have enough surveillance. We have cold spots. We don't know where the disease is. We don't know what it's doing. We don't know where the next outbreak is going to come from because we're not able to track everything. So surveillance gives us a good idea of where we need to focus on. Forecasting. What is the need? As manufacturers, we're sometimes asked, what is our capacity? In many ways, it's the wrong question. Because we can increase our capacity, we can build capacity, but capacity is dependent on demand. If we don't know what the demand is, we don't know what's needed for the future, and therefore we can't build for it. And that's where forecasting comes in. If we have strong forecasting models, if we know from a country that they're going to need vaccine next year and what volume, and then the year after, then we're able to do something about it. What is your objective? Are you trying to eradicate disease? Or are you just trying to control it? Or are you trying to remain free? All of these things, understanding your objective, all of these things are important. And involvement, engage the stakeholders all across the board. And that means talking to manufacturers like us. So we understand, again, going back to that forecasting and your need, if we're engaged in the discussions, we're able to think about what we can do to help and support rather than just supplying a vaccine. There are plenty of other areas that we understand or are able to engage with and help. But engaging with people at the local level, engaging with village elders, engaging with the local vets, all of these things, because when we don't do that, we can disenfranchise people, we can drive them away. One example is local vets sometimes see government programs as taking away from their income. If they see free vaccine being brought out, they're thinking this is something they then can't sell. Or if you get a disease under control, then the local vets suddenly don't have a stream of income because they're no longer treating for that disease. So they need to be engaged and brought into the overall picture. What are the possible approaches to prioritization? And there are quite a few now that we've understood. And some of these, the first two of these, are thanks to Dr. Tiziana Lembo and Dr. Divine Ekwem, University of Glasgow. Some work we've been doing with them in Tanzania has shown some really interesting work. One of those is by risk area. And that's been discussed before a number of EuFMD meetings and other FMD research meetings where we've looked at where the risks are and understanding the nodes of connection. So do you focus on vaccinating in local villages where the animals aren't really changing very much, it's fairly static, or do you look at the centers where the animals are brought for trade and purchase and sale, and that's where the disease is really mixing and starting to spread up? So maybe that's where we need to be focusing our vaccination efforts rather than now in the villages where there's not so much disease driving around. Look at where it's being brought in from rather than where sometimes it seems like some of the disease is actually happening. So tracking back to understand those nodes of connection. But we also could look at circulating strain. This seems, especially in Africa, really possible. That what we're seeing is the different strains have patterns, and you can see waves of certain strains traveling across the country. And that gives us an opportunity to vaccinate ahead, to create barriers. So rather than necessarily trying to go with trivalent, quadrivalent vaccines, which are packing everything in together, maybe we should be focusing on what disease do we think is coming and having a monovalent approach, but vaccinating targeted for what is actually coming. We can look at the time, focus on when movement is occurring. When are we most likely to be able to see those diseases, and when do we know those diseases are actually circulating around? Well, that's a good time to focus on. Now's the time to vaccinate. Well, 2 weeks before, 3 weeks before, so animals are immune and therefore we don't spread the disease when they're actually moving. And that movement can be driven by trade, it can be driven by festivals, it can be driven by the weather, but every region has its own patterns. Those patterns do change with time. We need to be aware of those and see. And then also focusing and prioritizing by the vaccine, by the quality and by the type, looking at what is it doing, what's it trying to get to. So in summary, there are limited resources, but what we see is prioritization is definitely cost effective. What you need to think about is what is your objective? And then once you've got that, then consider various solutions and communicate with all the stakeholders in order to get the results that we require. I hope that this was helpful and stimulates some thought, and I look forward to discussing it further with you.
Thank you so much, Alistair. That was a very fascinating talk. I really appreciate that. We are still trying to solve the problem, the technical problem that we have with our keynote speaker, Dr. Hodlet. Meanwhile, we decided to go ahead with the recorded presentations. Before I introduce the next speaker, may I ask you again, please post your questions to the presenters into our EuFMD discussion forum. You can see the number of presenters there, the name of presenters there, and you can post your questions under their title. So the website is again eufmdvirtual.com/forum. Please post your questions there. Thanks very much. I would like to introduce our next speaker. Our next speaker is Dr. Rukaya Horchani, from Director General Services of Veterinary Medicine, Ministry of Agriculture, Republic of Tunisia. The title of Dr. Khorchani's talk is Evaluation of Human Resources Needed and Comparison with Human Resources Available to Control Outbreaks of Foot and Mouth Disease in Tunisia using Emergency Vaccination. We will listen to the recorded presentation by Dr. Khorchani.
Hello everybody, my name is Rukaya Khorchani. I'm a veterinarian. I'm working in Tunisian Veterinary Service. I'm talking during this presentation about evaluation of a human resources needed and comparison with the human resources available to implement emergency vaccination in case of FMD outbreaks in Tunisia. The study were conducted in collaboration between Tunisian Veterinary Service and ANSES France. After a short introduction, We will speak about the methodology and the main results of the study, and we conclude at the end. It is important to focus in the FMD epidemic which took place in Tunisia, the organization of veterinary service, and the objective context of this study. The last FMD outbreaks in Tunisia took place in 2018-2019. A total of 14 outbreaks were notified, mainly in the center and the northwest of the country, and the cases were in cattle and small ruminants. The veterinary service is in Tunisia is organized as a central department, the General Directorate of Veterinary Service, which include the Animal Health Department, Animal Product Control, and Border Control Department and Certification, and have the role of making strategy, planning, and evaluation. The Animal Production Department, which include the Regional Veterinary Service under the Regional Commissariat for Agriculture Development in 24 governorate, and have the role of development and control. The main objective of the study was to determine human resources necessary to manage FMD outbreaks in Tunisia, then determine available resources for each governorate, and finally to compare and determine the gap between human resources necessary and human resources available. What about the methodology of the study? A human resources requirement grid was developed by combining the tasks provided by the national response plan to manage FMD outbreak in Tunisia. We only consider the tasks carried out in the episode of FMD in 2014 in Tunisia, such as investigation of suspect cases, implementing vaccination, implementing point of disinfection. Then we have a workload per task and skill. Field surveys consisting in interviewing animal health manager in 24 governorates in Tunisia to quantify the number of human resources available to manage FMD outbreak in each governorate. We quantified the number of veterinarian, technician, administrative agent, and estimate the number of herd in each governorate. We then calculate the gap between the resources. We calculate the time needed for all tasks. We calculate the time needed for emergency vaccination, and the approach of time needed for emergency vaccination was validated by observing the duration of emergency vaccination carried out in the episode of 2014. What about the results of study? As you see in the table, we have 3 task group: outbreak management tasks in field, and the organizational tasks such as crisis unit, communication, and implementing emergency vaccination. We have the duration of each task in hour and the amount of skills such as veterinarian, technician, driver, administrative agent. We estimate the human resource available in each governorate by the field survey. The human resources can be veterinarian, technician, administrative agent, appointed private veterinarian, and we calculate the vaccination team, such including the driver. we have considering 8 scenario in estimating the duration of emergency vaccination. The scenario 1 by using public resources. The scenario 2 by using maximum level of public resources. Each team vaccination compound veterinarian or technician. The scenario 3 using public resources and appointed private veterinarian. The scenario 4 using maximum level of public resources, then appointed private veterinarian. And the scenario A and BA, the vaccination is generalized, vaccinated all species. And this is the scenario B, cattle targeted vaccination. The chart A shows the number of herd in each governorate. The chart B shows the number of veterinarian in each governorate. And the chart C shows the duration of emergency vaccination depending on the scenario. On the scenario 1A, using only the public resources to manage FMD outbreaks, we have 12 governorate which complete the emergency vaccination in more 6 months. In the scenario 4B, using the maximum level of public health resource, public human resources, and appointed private veterinarian, all governorate can complete vaccination in less than 2 months. So we have, as a result, the total workload per task and skill with field skills, field tasks, organizational tasks, and the time dedicated to vaccination, which is the major part of workload. We have also the time needed for emergency vaccination, which varied according to the scenario, using public resources or public and appointed private veterinarian. We have seen that using private vet limit the time to less than 4 months for all the country. In conclusion, developing a resource requirement grid and evaluating available resources in each governorate help us to compare resources to manage FMD outbreak in Tunisia in each governorate. We conclude that implementing vaccination is a major part of time and the predict emergency vaccination campaign duration depending on the scenario and the strategy of vaccination. Vaccinate all herds, cattle and small ruminant, or vaccinate only cattle. This tool could provide support to veterinary service on managed FMD outbreak. Finally, I would like to thank ANSES, EuFMD, DGSV, DGL, CRDA to support this study. Very much.
Thank you so much, Dr. Khorchani. I think the problem with our connection to Dr. Hodelet has been resolved. I would like to know if that's the case. We can go ahead with live presentation by Dr. Khorchani. By Dr. Hodelet. Otherwise, we continue with recorded presentations.
Je ne l'entends pas. Buddha, can you hear me?
Yes, yes.
And can you do it with your head? Because
yes,
okay,
we can hear you. We can hear you.
If you all can hear me, we're gonna do it like that. Because apparently, if you see me and you hear me, that's all it takes. Because I cannot hear you. I am extremely sorry for these technical difficulties, and I propose to go ahead with my slides.
Sure.
Thank you. Whew. Dear colleagues, next slide. Next slide. This picture was taken only 2 years ago. It feels like an eternity, doesn't it? Look how close we were to each other. Look how we all had smiles on our faces. Now we can see our smiles. We cannot see our smiles because of the masks we wear. I'm surrounded by colleagues and they all wear masks. 2 years ago, the EuFMD Open Session was still focused on FMD. And we were just starting to address the issue of vaccine security. Today, I will dive into one of the solutions that exist to address the lack of vaccines, and it's called vaccine banks, or stockpiles, or strategic reserves. Whatever you want to call it, the model is not very widely known, and there is always an element of secrecy because of its strategic nature. So I think it'll be useful for me to share our experience in this forum. Next slide. Two years ago, our dear colleague George Matlow was still with us. George was a good scientist, a good man, and a friend. Dumela, Kego Gopotse, my friend. Rest in peace, George. We haven't forgotten you. Next slide. So I'll start with a few words on vaccine security. Vaccine security is the timely, sustained, uninterrupted supply of affordable vaccines of a short quality, according to the UNICEF WHO definition. In practice, when are quality vaccines not available? Well, first, of course, when they don't exist. That was the case for African swine fever or SARS-CoV-2. And that was the subject of the other keynote presentation. But also when there are no stocks readily available to serve the demand in time. So this point can be addressed by banks. Banks are a truly unique business model that can address sudden, unplanned demand for quality vaccines. Next slide. To illustrate the specific challenges of the supply chain for FMD vaccines, I will mention this. We have to manage a highly variable demand with constant competition between regular demand and peaks that can be due to outbreaks, but not only. Sometimes they are also due to unexpected funding that becomes available. And unfortunately, there is no idle manufacturing capacity that is sitting out there ready to respond to these surges. Anywhere, there are no stocks. Or when they are, it's not the right strain. So you have another issue. Sales forecasts are often unreliable because the market is mostly based on tenders or on orders with unrealistic expectations. For example, the vaccine shall be delivered within 10 weeks, like in some FAO tenders, when in fact it takes several months to produce a dose. So banks remain the only available solution to serve rapidly a sudden request for vaccine. The examples I'll take in the rest of my presentation are mostly based on FMD vaccines, because this is where the problem is most acute. But the conclusions actually apply to all vaccines. Next slide. Now a little bit of history. It's a 50-year story. Who invented the banks? Sorry, my Swiss colleagues. The idea actually came from England. I have to give back to Caesar what belongs to Caesar, even if it hurts. The first mention of vaccine strategic reserves was made in the UK by a commission that reviewed the lessons of the great epidemics of 1967. In the 70s, a famous French company demonstrated that you can store FMD antigens as a concentrated and frozen form. In 1979, the first antigen bank was put together by a group of countries for North America. In the 80s, there was an international vaccine bank, which did not survive the 90s. And banks really developed in the 90s after European countries stopped vaccination. Next slide. So who can hold banks? Obviously, countries that do not have the disease on their territory and may want to be ready in case it is introduced. But not only, it can also be useful for endemic countries. For example, for exotic serotypes. Middle East countries might see an interest in building an FMD bank with African strains or with Asian strains in case there's a spillover from neighboring pools. Or they can serve as a buffer stock of vaccine in case there is a sudden increase in demand. They can be national or regional banks, and actually mutualization makes sense. Who supplies the bank? Very few manufacturers do, especially for FMD. At Boehringer Ingelheim, we hold banks for rabies, FMD, and blue tongue. Other manufacturers hold banks for lumpy skin disease or PPR vaccine. For FMD, there are a few of us because it takes a broad portfolio of strains to build a comprehensive bank ready to answer any type of outbreak. And it also takes a very specific expertise and investment in that model. It takes a dedicated team and dedicated processes to be able to offer banks. Next. So the question everybody might have is, what's in a bank? Well, it's nothing like what you see in this picture. Next. First, let's take the example of the FMD production process to highlight the different types of banks. To make an FMD vaccine, you need to grow cells, you need to infect them with virus, you need to inactivate the virus, and then to purify it. Next. And all of this is biology, so it takes time, roughly 4 months from start to inactivated antigen. On the other hand, it takes only a few days to mix this antigen with adjuvant, fill it into bottles, and package it. Next. So you can store the active ingredient. This is an antigen bank. Or you can store the finished product. This is a vaccine bank. For FMD, both are possible. Next. So banks can be final, filled vaccine stocks, stored in bottles, labeled at +4 degrees, or they can be bulk antigen, which is kept frozen at very, very low temperature, usually on the vapor of liquid nitrogen. From a manufacturer's point of view, vaccine and antigen banks are not mutually exclusive. You can hold a combination of antigen banks and vaccine reserves. Some countries actually choose this option. And you can also devise smart replacement schedule at the end of your shelf life to minimize wastage of product. Next. There are advantages and drawbacks to both models. I'll mention just a few. Delay. This is only one aspect. A vaccine is readily available, whereas an antigen has to be finished into vaccine and released before completion of the quality control tests. Shelf life is another important parameter. Antigens are very stable. They can be stored frozen for several years, whereas vaccines have a relatively shorter shelf life, between 12 and 24 months, depending on the vaccine. Number of strains, that's an important one. With the antigen model, you can store each of the most important strains and decide at the last moment which one or which ones you want to put in your vaccine. With the vaccine model, composition is fixed. You can't change it at the last minute. This is okay for rabies, but this can be problematic for FMD. And finally, location is another difference. You can store vaccine at +4 degrees in multiple locations, in your country if you wish, whereas antigen has to be stored at the manufacturer's premises. Next. After production, the antigens are stored in one of our facilities at Purbright in the UK or in Lyon in France, and they're dedicated to a specific customer. There is restricted access to the vault. Only 1 or 2 people have access. And tanks are, of course, equipped with a whole range of equipment to ensure the security of the antigens. And we have to conduct periodic tests to ensure the stability of the antigens. We test 146S content on a yearly basis, and we do serological potency at time 0, 2.5 years, and 5 years. Next. Now, let's look at what a bank activation looks like in practice. Let's say there's a clinical case of FMD in a European country. Next. The situation is described in Article 50 of the European Union Directive on FMD. Emergency vaccination can be used if there's an outbreak in one of the member states and it threatens to become widespread, or if there's a risk of spread to another member state. Next. So here are the steps of a bank activation. Next. First, the country has to place the order. I receive a call, me or one of my colleagues. It's usually on Christmas Day or New Year. Anyway, it seems to always be during my vacation. And I check that all the information is available. The order has to mention the strain or the strains, number of doses, size of the vials, delivery details, an early release certificate, which is a statement from the chief veterinary officer that it is okay for us to ship a product that has not completed its quality control tests, and a timeline, how fast you want it. Next. By then, we stop everything we're doing. It's code red at BI, all hands on deck. We thaw the antigen, blend it with the adjuvant, and fill the vaccine into the bottles. Next. Then we package it, put it in boxes under cold chain, and next. And then it's on the, next. It's on the road or in the air, depending on the destination. Our commitment is that vaccine is always available at factory gate in less than a week. Depends on the contract. Next. So this is exactly where it would happen. These are some pictures from our Purbright site. You can see our blending and filling lab. We can formulate 2 types of vaccine, aqueous or oily. We can formulate several sizes of vials, and we can label in any language. Next. Vaccines are then put in boxes with ice packs, of course, and temperature loggers, because it is, of course, critical to maintain cold chain conditions from our factory to the point of delivery. Next. And finally, a very important point. In this emergency situation, we have no time to wait for quality control results. But this is a situation that is actually covered by the European Pharmacopoeia, which says that in cases of emergency, it is okay to release a batch and ship it before completion of the tests, provided the sterility test has been carried out on the antigen and on all the components of the vaccine, and that potency has been tested on a small-scale vaccine representative of the final batch. So in summary, banks are a complex process. It takes time to build such expertise. You need a dedicated team, dedicated processes, and dedicated equipment and supplies too. You need not only store the antigens, but also adjuvants, bottles, labels, etc., etc. And it's a commitment because you might have to drop everything you're doing if the request comes. For example, if there's already a batch of vaccine being prepared in one of your formulation tank, you might have to throw it down the drain to make some room for your bank. And it takes practice. We have several activations every year, and we have never failed to honor our commitments. I should say, of course, because an antigen bank is like an insurance. You hope you never need it, but when you do, it has to deliver. Next slide. Now, to go back to a point I made earlier, banks can really be useful in multiple environments. Next. Today, the antigen bank model is mostly used in FMD-free countries. We see initiatives going towards mutualization of banks. There was an international vaccine bank in the 80s and 90s. The European Union and North America have mutualized banks, and the Quads have also pledged to help each other. There are downsides to these models. First, some financial considerations. At the end of the storage period, some valuable antigen stock often may have to be destroyed. And also, unfortunately, policies and plans are not always there to allow vaccination. Preferred options often remain 1, stamping out, and 2, vaccinate to kill. And I will also mention that allocation rules for mutualized banks are not always clear. The rules to decide which country would get the doses first and how many are not always clear to us. But that's not up to us to decide, of course. Next. As I said earlier, banks are also a good model for endemic countries, either as a stockpile that's ready to respond to a sudden surge of demand, or to an outbreak with an exotic strain. But it can also be a way to trigger, to encourage vaccination in developing countries using pilot vaccination campaigns to demonstrate their benefits. It could start with regional banks. There are some. They're usually funded by international organizations. There's an OIE bank for Southeast Asia, was funded by Australia and then New Zealand, but unfortunately this one was stopped. There is a rabies vaccine bank funded by the OIE and the WHO, and there's a PAHO rabies bank for South America. But really, what we need is to build sustainability in the model. For countries in the early stages of the PCP, donor funding is necessary, sure. But afterwards, I believe there should be greater support beyond funding towards putting in place the infrastructure for vaccination. And this should be coupled with economic analysis. So you can use these pilot campaigns to generate some hard data showing the effectiveness and the cost benefits of vaccination. And once the infrastructure is in place, countries may realize that vaccines are only a fraction of the cost of a control campaign. And they may continue on their own. So next and last slide. As a conclusion, antigen and vaccine banks are one of the solutions that can help you reach vaccine security. Today, they're mostly used by disease-free countries as an insurance, part of their risk mitigation strategy to minimize the consequences of an outbreak. But they could also help minimize vaccine shortages in endemic countries. They may seem costly to put in place, but they guarantee access to quality vaccines when needed. Thank you very much for your attention. I'm not sure I can hear your questions, but I'll stop there. Okay. I can read a question if you like. I received one on WhatsApp. Okay, thanks. Can you describe the work that decides how to include the correct mix of FMD serotypes and lineages? So this is work that is done through the activities of the World Reference Lab. It is based on a mix of epidemiological analysis, looking at the circulating serotypes and lineages worldwide. For a country, then it would be the question of deciding which region is more of a threat for them, from which the introduction could come. And then the other part is vaccine matching, deciding which vaccine strain works best to control against these potential threats. And matching the two, you come up with tools. We have developed our own tools at BI to give this visualization for customers. The World Reference Lab and EuFMD use a tool that is called Pragmatist that also works well for that purpose.
Thank you very much, Pascal Villeret. It was a very clear presentation, and we hope that you can join us later for the discussion in the round table after eventually the problems have been solved. And then Buddha will take us to the next presentation.
Thanks very much, Sten, and thanks very much to Pascal for his presentation. I'd like to introduce our next speaker. This is going to be a recorded presentation by Dr. Amin Asfar from the Pirbright Institute. The title of Dr. Asfar's presentation is Development of a Peptide ELISA for specific detection of foot and mouth disease virus neutralization antibodies. Can a test in this format replace VNT? I would like to ask my colleagues to broadcast his presentation.
Hi everyone, my name is Amin Asfar. I am a virologist at the Pirbright Institute and a teaching fellow at the University of Surrey. Today I'll be talking to you about the development of a peptide ELISA for specific detection of EuFMD antibodies, and answer this question here: can a test in this format replace VNT? If we talk about the structure of EuFMD has a icosahedral capsid, which forms of 4 different viral proteins. On the surface of the capsid is VB1, VB2, and VB3. And if you have a cross section in the capsid, you'll find another protein here called VB4, which is important for virus stability. Protection against the virus is antibody mediated through neutralizing antibodies. Hence, there are 5 well-known and well-characterized neutralizing antigenic sites for serotype O, and there are equivalent antigenic sites for other serotypes as well. One of the most important structure of EuFMD is GH loop of VB1, which contains the immunodominant epitopes, mainly site 1 and site 5. GH loop and immunodominance. GH loop contains a highly conserved RGD motif that mediated binding to integrin receptors. As you can see here, this is the RGD motif. The GH loop itself is a flexible loop that can adopt 2 main conformations, either up conformation or down conformation. If it is the up conformation, it forms a series of overlapping linear epitopes forming site 1. If it is in down conformation, it forms a single conformational epitope forming antigenic site 5. When it comes to the problem which we are trying to solve by developing this new assay, is across the activities. In a very big experiment that's been conducted by WRL, they found that a significant cross-reactivities were observed using serotype-specific kit against sera from different serotype, using 5 kits either in-house or commercial. Just one quick example: if we have serotype O sera and we'd like to test it against different serotype kits, you'll find that serotype O sera will behave well in case of a homologous kit, whereas if you would like to use the serotype O sera with serotype A kits, there is a cross-reactivity of 46%, which is not really good. So the take-home message from this experiment that there is a huge probability that if you use one serum sample against different kits, there is a cross-reactivity that could reach it to 50%. That's why we are trying to develop and improve methods for serology of EuFMD. The current serological methods, which are golden standards, is VNT. As we all know, VNT is highly demanding and requires high containment facilities. How VNT works: we have a live virus and cells that support the replication of the virus, which express integrin receptors. The virus will bind to the receptors, enter the cell, and start replication. However, if we have the sera specific to FMDV that will inhibit this binding, hence the neutralization of the virus will happen and we'll be able to detect the titer. As I said before, the VMT is highly demanding and required high containment facilities, which is rarely found in developing countries. That's why we are trying to develop and improve methods, mainly ELISA and other immunoassays. In case of ELISA, we are targeting the native antigen, which is actually should be expressing the surface-exposed epitopes, of which it is actually neutralizing epitopes, and some non-neutralizing epitopes highly conserved. If you, however, due to low pH during the processing of the antigen or during the processing of ELISA, this antigen can be degraded into the pentameric form, which show the inside of the capsid, which has certain non-neutralizing epitope, which is highly conserved. When you add or test the sera, what will happen? We will have two responses. The first response is type specific, and the second response will be a cross-reactive response, and sometimes outlayer the specific response. Hence, we are actually observing this high cross-activity in the current commercial available kits. So I came up with the idea of using a single linear neutralizing epitope that could be used for a specific section of FMDV antibody, and whether or not this approach can replace EMT. We have some tools developed over the years. One of them is the GH loop peptide, and the GH loop is specific neutralizing monoclonal antibody as a control, and the cross-reactive sera, which is a highly cross-reactive. As you can see here, this is serotype O kits against serotype A sera. The sera here are highly cross-reactive, 4 out of 5 kits or 5 out of 5 kits, and some controls as well, either a homologous sera or a negative sera as a control. The peptide ELISA format, which I am developing, is based upon a single site. One of the neutralizing antigenic site in the GH loop peptide is site 1. You coat the plate with this peptide and then come with the sera and develop with the secondary antibody and the GMP. So the first data which we have to support the validity of this approach, as you can see here, this is two different ELISA, one using peptide ELISA and the second one using O-Manisa covalently caged capsid, full capsid. As you can see, the neutralizing antibody can equally recognize either the peptide or the full capsid, which is a good news. Hence, I have started to do another experiment, which is again based upon ELISA, integral ELISA coated plate, capsid will bind, then block with D9, which is neutralizing antibody recognized GH loop, and then add the sera to see whether the sera will bind or not. And the sera actually did not bind. Hence, you develop and see the first result is if you block with D9, the sera will not bind, indicating that the response which we are seeing in the full capsid is mainly targeting the GH loop as an immunodominant epitope. However, if you use other monoclonal antibodies which are non-neutralizing, as you can see here, if you compare it with no antibody treat, you will find there is no actually inhibition, which again another use. Then I've tested some of the cross-reactive sera, which has been used again using the full capsid. As you can see here, at high concentration of the sera, you'll find lots of cross-reactivities here, even outlayer the homologous response. And we believe that this cross-reactivity is due to low ability antibody. When you actually dilute the sera, you can see that this cross-reactivity went away. So I want, however, if you would like to actually develop a spot test or spot ELISA, you need a high concentration of the sera. That's why I came up with this idea using the peptide ELISA and tried all this cross-reactive sera. You can see here at 1 in 50, there is no cross-reactivity, and you have a good response of the homologous sera, which is again good news. Then I asked myself a question whether or not this approach can be used in terms of vaccine matching, if you would like, or there's any differences in responses if you have different sera or serotype O within serotype O, but different strains of the viruses. As you can see here, in this is a different peptide concentration, one dilution of the sera. You can see there is a different vary in response to different sera raised against different serotype, strains of the serotype O. Just if you zoom out of one particular concentration of the peptide, it will be very clear that serotype A, which was cross-reactive, is not cross-reactive anymore. And here you can see a variability in response. So this variation could be due to sequence variation in the fingerprint of the peptide used, or due to quantitative difference in neutralizing antibody level in VMG. And that take us to next question. Can a test in this format replace a VMT? So we compare our data using the peptide ELISA with the already data available for liquid phase-blocking ELISA and VMT. As if you compare between these two tests here, you'll see in terms of the profile, a perfect match with one advantage of the peptide ELISA. It got rid of the cross-reactivities. Whereas if it comes to VMT, the profile is more or less the same. However, one of the sera here is not cross-match with the peptide ELISA. So the conclusion from this slide is that peptide ELISA OD is positively correlated with liquid phase-blocking ELISA and to a certain extent with virus neutralization test, but more data is needed to confirm this observation. With this, I would like to conclude that the cross-reactivities has been seen in all the commercial ELISA kits available, and we have tested. This cross-reactivities is due to internal epitopes and the conserved service exposed epitope. GH loop could be one good candidate for an improved methods of serology and detection. For the GH loop, the type is correlated to a certain extent with VT. The future work will be testing more sera and also testing more GH loop peptide representing different serotypes and try to correlate that with the VMT and protection. With that, I would like to acknowledge the people who contributed to this work, also Eisler, Amy, and Santina for providing the monoclonal antibody. And I'm happy to take any question.
Thank you so much, Dr. Asfar, for your presentation. Of course, this was a recorded presentation, and we will get questions in the round table. I should say all our speakers will be with us in the round table at the end of this session to answer your questions. So please keep sending your questions in the forum that we will channel them to the speakers this afternoon. Without wasting time, I would like to introduce our last speaker of this session. Our last speaker is Dr. Izasur. Dr. Izasur is a lecturer at the University of Nigeria. The title of his talk is Intranasal Vaccine Application Methods Influence the Immune Responses in Goats Following Intranasal Post-Herpes Ruminant Vaccination. Clinical, pathological, and immunohistochemical findings. Please broadcast Dr. Izasur presentation.
Good afternoon, ladies and gentlemen. My name is Dr. Chukwunonso Kenechukwu Ezaso of the Department of Veterinary Pathology and Microbiology, University of Nigeria, Nsukka. I briefly want to talk to you about my research. It's an ongoing research, and it's titled Intranasal Vaccine Application Methods Influence the Immune Responses in Goats Following Intranasal Pes Dispetti Ruminant Vaccination, Clinical, Pathological, and Immunohistochemical Findings. As many of us may already know, PPR is a transboundary and highly contagious viral disease of small ruminants caused by morbillivirus, PPR virus. And like many viruses, vaccination is the major method of control of this disease, especially in the third world countries where the stamping out policy cannot be implemented due to obvious economic reasons. PPR vaccines are commercially available for subcutaneous injection. However, experimental intranasal administration of PPR vaccine has been successfully described in goats by numerous researchers. And this has pitched it against the conventional method as having more advantage in terms of general costs, in terms of user friendliness, in terms of ease of administration in field conditions, and thus may be most effective for mass vaccination campaigns. However, little is known about the influence of intranasal pes ruminant vaccine application methods on the induction of immune responses in goats. In this study, we used 20 male West African dwarf goats between the ages of 6 and 9 months, and they were divided into 4 groups containing 5 animals each. We used the live PPR vaccine, Nigeria 75-1 strain. Group A was vaccinated intranasally using a dropper. Group B was vaccinated intranasally using the nasal spray device.
I think we have lost the sound of the presentation. I think we have lost the sound of the presentation by Dr. Isezer. We are trying to find a solution for it. I do apologize for this technical problem. We will be resolving this issue. Just to have an overview in case you have missed this morning sessions while my colleagues are resolving this issue. We focused on basically how to allocate the resources for vaccination campaigns in the morning. So I will provide a more detailed recap at the end of the session. But in case if you have missed that session, the recording will be available on our website, so you can go and watch it later on after this session has ended. On that note, I'll wait for my colleagues to let me know if the presentation
Dose
will be
of
ready or
the intranasal through day 28. However, it did not differ significantly with those of the intranasal spray group at probability of 0.05. And also the antibody titers of intranasal drop group was also higher than those of the subcutaneous group by day 7. However, by day 14 through to day 28, those of the subcutaneous groups were higher, though there was no significant difference between the groups. Histomorphological examination of the lungs showed multiple bronchus-associated lymphoid tissues in the pulmonary parenchyma of the lungs taken from the group that were vaccinated using the nasal spray technique. And these samples were subjected to PPR immunohistochemistry, and we all discovered that the PPR viral antigens were present in the lymphoid cells of the bronchus-associated lymphoid tissues. Then PPR antigen was also detected via immunohistochemistry in the lymphoid cells of the spleen and the mediastinal lymph nodes in all the vaccinated animals after 28 days of vaccination. Findings of this study showed that PPR vaccination using a nasal dropper may result in a higher induction of PPR-specific antibody levels than those vaccinated via the intranasal spray method. However, the animals that were vaccinated via the intranasal spray method may have resulted in the deposition of aerosolized vaccine antigen deep into the lower respiratory tract and resulted in the development of bronchus-associated lymphoid tissues in the lower respiratory tract. And previous studies have shown that bulbs plays an important role in local pulmonary immune response. And some researchers have also reported that it is important in the protection of the lungs against PPR-induced capillary pneumonia. The results of this study also suggest that intranasal administration of PPR vaccine using the nasal dropper was able to induce a strong systemic immune response, but little or no local immune response in the lower respiratory tract, which conversely was better induced following intranasal PPR vaccination using the nasal spray. Thus, the nasal spray method of intranasal PPR vaccination may hold greater potential for pulmonary protection against the pneumonic form of the disease, much more than the intranasal PPR vaccination using the dropper method. However, this is subject to experimental research, experimental infection, to see whether the antibody response produced may be protective enough. And that is what we're going to do in the next study. Hence, the choice of application methods for what we have seen in this study for intranasal PPR vaccine delivery is very, very important and should be carefully considered in the development of intranasal PPR vaccines. Thank you very, very much for listening.
Thank you very much, Dr. Azor. We apologize for the little problem we had, but we can come back to that in the round table. Now we will, in a minute, start the round table. But before that, I would like to thank all of the presenters. I think you've created some very clear and nice presentations for all of us, and I've enjoyed it a lot. So we will need 1 minute to prepare for the round table, so you can stretch your legs, go around the table and come back, and we'll be ready. See you. Hello, welcome back. Is everybody ready?
Sure.
Good. And Pascal, you have sound this time?
Yep, the
I
sound is
do.
okay.
Oh, this is going to be great. Okay.
Yes.
Yes. And so we will be ready now. And again, Dr. Isesor, apologize. We apologize for the little technical problem during your presentation. Is there anything you would like to add that was fell out of your presentation before we start? Then you can maybe, if there's something you would add of something we missed because of the technical problem.
Okay, no, I think the technical guys did a good job in mitigating the problem.
Okay. Well, then I think I will simply move on to the first question to you. Was there any notable difference in the ease or tolerance of the three vaccination approaches?
Okay, I think the question says ease or tolerance. If I understand correctly, there was actually a notable difference in the ease of administration of the vaccines, especially the intranasal group, because in administration of the vaccines in the international group, little or no assistance was actually needed. It was just get the animals and apply the spray or the drop. But for the subcutaneous group, there was need for an assistant to restrain the animals before administrating the vaccine subcutaneously. So there was actually a notable difference in ease of administration. And for the tolerance, I think there's no difference between the groups for the tolerance of the vaccination approaches.
Good. And you mentioned in the end that you are going to measure the serological response. But apart from that, what's next to follow up on your work with this interesting and potentially efficacious vaccine?
Yeah, the next plan actually is to repeat the study and of course challenge with a live PPR virus in Nigeria 75 strain, which is the one that is available in our environment. So that's what we are hoping, because at the end of when we do that such studies, that's when we can actually say whether this vaccination approach is going to be protective enough, because that antibody responses were generated doesn't necessarily mean that there's protection. Okay, so it's when we do the challenge studies in the nearest future, if not for the COVID-19 lockdowns and the challenges with the year 2020, we would have at least gone quite far with this study. But that's it.
Can I just come in there, Sten?
For sure.
Just a quick question. Do you expect the duration of immunity to be the same, having topically administered the vaccine? And had you considered doing a prime boost strategy, so you prime by a local route and boost intraperitoneally?
Okay, well, we can't really say anything concerning the duration of immunity at the moment, because we've not yet done a challenge study, which when we confirm first that is protective, then we can go ahead and check for the duration of immunity, and also check if it's if effective in maybe pregnant animals and all that. So these are still things that we are looking at doing in the nearest future. I hope I answered the question.
Mm-hmm. Yeah, okay. Next question for Dr. Amin Asfar. If the future work on development and validation of a peptide ELISA for specific detection of foot and mouth disease antibodies is promising, how far away in time are we from using this really as an alternative to VNT?
I think this is a very good question. I have to mention this is only preliminary data. So we have to spend some time actually validating the data and testing lots of different sera from different settings as well, just to come with this conclusion. However, I think from the preliminary data, I sort of very confident that it will go somewhere. In terms of timeline, maybe in the next EuFMD or so, maybe I'll come up with a confirmation for the data.
Thank you. Any other questions from the or the participant here to Dr. Amin Asfar?
Could I just ask, how long is your DH loop peptide, and have you thought about structuring that peptide in any way?
Yes, I mean the GH peptides are around 30 amino acids in total. But again, I think this is a good question because I'm trying to try to make a cyclic peptide that may be better show the conformation of the epitope. Hence, I would expect a better response as well.
Yeah.
Okay, thank you. Then I have a question for Dr. Khorshani. To what extent, what specific way have these findings influenced the FMD response plan in Tunisia, specifically with regard to vaccination?
Thank you for the question. The update of FMD response plan in Tunisia is in progress, and surely we will consider this result, in particular in emergency vaccination. Now we estimated the time needed for emergency vaccination in Tunisia. So we'll adapt our strategy in emergency vaccination depending on resources available in each governorate and the objective of emergency vaccination, the time objective of time vaccination. If we need emergency vaccination in 2 months, we'll adapt our strategy, maybe vaccinate only cattle, not the small ruminant, or large premise cattle. So we'll adapt our strategy depending on the objective of emergency vaccination and available resources in each governorate.
I noticed that in some scenarios you're using private veterinarians. Have they been involved in the planning of this?
Excuse me, I didn't hear you.
Sorry,
Yeah.
I noticed that in your scenarios, some of your scenarios, you used private veterinarians. Have they been involved in the discussions of how to do this?
Yes, but in Tunisia, actually, we will use private veterinarian only in routine vaccination, not in emergency vaccination. So maybe using private veterinarian can limit the time of emergency vaccination.
Thank you. Yes, and any other questions from the panel members here at this point? Okay, then let's move on. I have a question for Dr. King. Dr. Kim, there are many questions for Dr. King, I can say. And I'll find it. Sorry, I'm a little bit. See if I can find it. Yes, from Dr. Shankar Yadav to Dr. King. He thanks you for a very interesting presentation. Regarding the vaccine quality, I wonder how a client, be a veterinarian or technician or farmers, be better informed whether a vaccine available in the markets is of high or low quality. Is there any possibility to eliminate the production of low quality vaccines?
That's a challenging question, that one, to be sent out. I think there's quite a few different things about that. I think the first, if we're talking about foot and mouth vaccines, foot and mouth disease vaccines, I think looking at the potency is my first area. That graph I showed of that work that we did with Wageningen, low potency vaccines are not doing the trick well enough. We need to be aiming for high potency vaccines. So that's one of the things to be able to look at. When governments are looking at buying vaccines, they can look at quality, they can talk to the companies who are making them, but also looking for evidence of GMP. It's not that straightforward because not all GMPs are the same. So you need to know that it's a good GMP, and I would say EU GMP is a very good standard. So that's very helpful. The good news is that the EuFMD is working on pre-qualification process. And I think that's well needed in veterinary medicine. We see it in human medicine, where vaccines for government programs get pre-qualification through the WHO and Gavi. And I think that's absolutely wonderful. We've missed that so much in veterinary medicine, but EuFMD are making those steps to actually start doing that, which gives a seal that shows that something is a good quality. I think it's important that just because it doesn't have EuFMD pre-qualification when that comes about, doesn't mean it's poor quality, but at least if you've got someone like the EuFMD giving pre-qualification, pre-approval, you know that that's something that you can trust. So that's going to be a game changer for us in the future, I think.
Okay, interesting. Then there's another question from P. Compton to you. He's interested in who would you suggest leads the prioritization process you discussed, the government, international agencies, or the private sector? Or does it depend on the context?
Thank you, Polly. I think that is everyone working together. And that's one of the things I did mention about us. Communication is so important. Surveillance needs to be improved. We don't have that level of understanding of what the disease is doing in a lot of countries. And I really do worry about cold spots where we don't see what's going on. That has to be done at a government level. Governments have to do the surveillance because no one else can get into that country and do it. But once we've got our surveillance data, really it's a case of us all sitting down together. It should be the governments, it should be the manufacturers, it should be the international organizations sitting down and looking at what is applicable for that particular situation. I started off saying there's no one answer because it would be wonderful if there was. Every country is different, and unfortunately, what we've tried to do in many cases is take a blueprint from one country and just drop that into another country and say, well, it worked there, so it will work there. That's not how we're going to get rid of FMD or many of these diseases. So we all need to talk and spend time. And then once that's done, we can understand what the individual needs are. In many cases, the private sector, the pharmaceutical companies can help with more than just providing a vaccine. I would go to the blue tongue outbreak in the UK, where we helped the UK government understand where the vaccine was being used and how good the uptake was to roll it out in different bands at different times. So we can all work together. So it's not one person leading, it's us doing it as a team. Thank you, Polly.
Okay, and then I have a question from Dr. Michael Francis. It's been encouraging to see how new technologies have been accepted for human vaccines against COVID, such as messenger RNA and Chatbox 1. How best can the veterinary community piggyback on this acceptance to accelerate the uptake of these technologies on the veterinary side?
Well, I think the good news is we don't need to piggyback on anything, because I think a lot of these technologies find their first application, as I was saying, within the veterinary field. If you look at pretty much all of the technologies now being exploited for use against COVID-19, they have already been tried and tested and often already licensed for use in veterinary medicine. So it's a frustration of mine that perhaps the human vaccine profession doesn't come and look more closely at what we do in the veterinary field. Indeed, in coronaviruses themselves, there are a number of successful veterinary coronaviruses out there for at least 5 different species using attenuated or inactivated strategies. And there's a lot to be learned from those vaccines in our attempts to address COVID-19. So I think we're very creative and imaginative, and I think a lot of new technologies really do get applied in veterinary fields.
That's promising and nice to know, really. I have another question here. There is sometimes a reference to a valley of death for veterinary vaccines transitioning from late phase development to registration and market. If you're aware of this reference, can you speak to any recommendations or strategies for successful navigation of this often challenging stage in the vaccine development process?
Yeah, okay. I've not heard it called a valley of death, but I think quite often research and development personnel have to really champion their vaccine to get it right through to marketing. And some vaccines can fail at a late stage, not because of problems with the vaccine itself, but because the marketing numbers decrease the closer you get to the market. So I think, you know, somebody who's developing a new product from scratch, they need a good plan. They need a way of taking it through development. As you get more and more data, you need to reinsure the market that the product meets the demands of the marketplace, and then really be persistent to push it through to the marketplace. But I do know of a number of successful vaccines that do fall at the last hurdle, and it is mainly because perhaps marketing numbers just drop away and nobody's going to promote and launch that particular product.
Yes, okay. And I guess I'm not very familiar with the production of development of new vaccines. So I speculate that many vaccines start at the universities, and then there needs to be a transition to the private sector. How do you see that?
No, that's absolutely true. I mean, I think a lot of new technologies do start within the academic field, and they're brought into the commercial sector. What I would say is it's important for these academic groups to interact with commercial groups as soon as possible so that they can help them and guide them in those early stages, because there are things they can do that could jeopardize the final registration of a vaccine without them realizing it at an early stage. But I think companies are very prepared to work alongside academic groups to help them and support them, and then to bring the vaccine development in-house. Really, when a vaccine moves from those early stages in terms of feasibility into full development, that's when you have to put the significant dollars against the vaccine to move it forward. And that's really where the experience from the larger veterinary pharmaceutical companies come into play, because they're very used to doing that and they're very familiar with taking it through that development pathway. What I would say in veterinary medicine is that because we're able to go into target species at an early stage, and because we're able to show protection in target species at an early stage, generally, when a vaccine goes from feasibility into development, it's got a very high chance of getting a successful registration, which isn't true within the human vaccine platforms. They generally go into clinical trials at risk, and as we've heard recently, a lot of them fail at that stage. But that's not true with veterinary vaccines. Most of them, the majority, will succeed once they get into full development.
Okay, thank you. Then there's another question for you here. The new human COVID vaccines cost between $4 and $40 a dose, whereas veterinary vaccines normally only cost between a few cents and a few dollars, depending on the species. How can we get recognition that the benefit risk evaluation is fundamentally different between humans and animals. And if we want some of the new technologies for animal use, we may have to accept different or lower quality standards, provided that the safety is not compromised.
Yes, that's an interesting question. I mean, I've worked in both the human and the veterinary field, so I've sort of seen it from both sides. I don't think we need to compromise on quality. That's the first thing I would say. And that veterinary vaccines are produced to a standard really equivalent to a lot of the human vaccines with GMP manufacture. What I would say is that within the human field, often you have a dedicated manufacturing plant for a single vaccine with all the support personnel. You have a greater margin in terms of what you can charge for your particular vaccine. And you have the requirement for further purification steps. I mentioned Chatbox. We're using that for both humans and animals. But what we found is that we can take that vaccine manufacture using the same seed up to a purification stage for a bulk harvest. And then we would use it in the veterinary vaccine. For the human vaccine, it undergoes a further few purification steps before it would then go into human trials. So there is more requirement for further purification. But I think there's a lot that the human industry could learn from veterinary medicine in terms of getting their costs down. And if you think about the biggest use of perhaps vector vaccines is in the poultry industry, where you are looking at just a few cents per dose. Those sort of technologies are still available to us in veterinary medicine.
Yeah, that's a good point. Thank you for that. And then there is a question for you, Pascal Videlay, from Melissa McLaws, and who was pleased with your presentation, as I were. You mentioned that vaccine antigen stock is destroyed if not used by the bank before expiry. Presumably, this would also occur for vaccines. This seems like a terrible waste given the limited global EuFMD vaccine supply. I was wondering if the antigen stock of vaccines is ever or could be redeployed for use in endemic countries.
Okay.
Yes.
You hear me okay? Okay. So you have two different situations here, whether you're speaking about vaccine banks or antigen banks. For vaccine banks, the vaccines, as I said, they have a relatively short shelf life. So they are rotated. There's a rotation of the stock of vaccine at their expiry date or slightly before. So they cannot be supplied to another country. Indeed, most of the countries require a minimum shelf life for vaccine that we supply to them so that they have time to use it. So for vaccines, it's really difficult. It's a rotation system. For antigens, it's different. At the end of the antigen bank, the fate of the antigens depend on multiple factors. Can be contractual commitments. There are some customers who say they want access to the antigen at the end of the shelf life, and they do whatever they do with it. And it depends also the shelf life of the antigen at the end of the period. If they have a specific shelf life and they reach that shelf life, for quality reasons, we cannot include them in another vaccine. So concretely, the antigens can be formulated into vaccines on behalf of the customer for donation, for example. That's the best case. They can be destroyed if they're at the end of their shelf life. That's the worst case. Can happen if there's no demand for this. Some strains have very low demand. SAP3, not a big market for that. Or they can reintegrate the manufacturer's stock, either for vaccine formulation for endemic countries or to keep stock until there's a need. As responsible manufacturers, we try to minimize this wastage as much as we can, of course.
Thank you. Thank you very much. Yes, then there is a question for Dr. Alistair King from Alto Decker. He says, you mentioned high potency vaccines should be used. Do you mean that the vaccine should pass the OIE criterion of more than 3 PD50 per dose, or would you suggest that the OIE increases this criterion to 10 PD50 per dose?
Gosh, Aldo, 10 PD50, that would really be pushing it up quite a lot. I was talking about 6 PD50. I think 3 PD50 is too low. Personal view, I've always said it, so no surprise to anyone who's seen me at these before. 3 PD50 is, I just can't think of any other veterinary vaccine where we go, oh yeah, we'll use something that's not quite so strong. We know what works. 6 PD50 cuts down on the transmission rate. It gets the immunity up quicker. It's 6 PD50 also will get us past some of the cross-reactivity problems. We've seen when we've got new strains, a 6 PD50 vaccine may work, even though it's not the matching strain, when a 3 PD50 doesn't. So I was talking about 3 PD, going up to 6 PD50, not using the 3 PD50, and that was the work that we did with Wageningen, was comparing 3 PD50 versus 6. Aldo, no surprise that you'd want to go even higher and push it up to 10, and really give us manufacturers a hard work to get really that level. I think 6 PD50 does the job very well, so that's what I would look at. Thank you.
Well, ambitions, they are always good. But I'm taking too much of the screen time here because Dr. Isaacson, he has a question for Mike, if possible. Dr. Isaacson, please.
Okay, Mike. Yeah, you in your presentation you made mention that reconstituted Chatbox 1 vaccine was stable at 30 degrees for a week, and I think 20 degrees for a month. So I want to know, is it possible that sharp variations in temperature may affect the potency? Why I ask this question is because because of the peculiarities in Africa, where you have a inconsistent power supply, you know. So and it's going to affect the storage, of course, and also in some places where it's usually very hot at night, assuming you want to store the vaccine on shelf, very hot at night during the daytime and very cold at night. So these possible sharp variations, is it possible that it can affect the potency of the vaccine?
Yeah, I think we would hope not. The adenovirus is actually quite a stable virus. And I have to say we're doing further work on it at the moment, but it's been interrupted, as you can imagine, doing this work with the Oxford group who are developing COVID-19 vaccines. It's sort of gone on the back burner a little bit. But what I would say is that we're looking at potency in terms of its infectivity, this particular virus in a permissive cell line. So it doesn't lose any of its viral titer when you move it between different temperatures. And we've also shown that when you've stored it at one of the elevated temperatures, say 20 degrees, it's still immunogenic when it goes back into an animal. So I think what we're hoping here is it gives one more flexibility. Although it may enter a cold chain, it doesn't mean that if it exits that cold chain for a short period of time, that it will become ineffective as a vaccine, which I think is very valuable. What I think is even more valuable is the observation that freeze-dried stabilized product is stable for up to 6 months at 30 degrees. And if you think about the debates with COVID-19 at the moment in terms of the vaccine stability, you could do that with a Chatbox vaccine for COVID-19. That would help the world and global distribution of that vaccine immensely. So I think there's still a lot to be done here, but I think stabilizing technologies will give people more flexibility under field conditions.
Thank you. And then the last question from Dr. Amin Asfar, also to you, Mike.
Yes, Mike. I'm interested in the ethical approval. So if you have an idea of developing a vectored vaccine of veterinary origin and you would like to apply it in human, how difficult it is to get the ethical approval?
Sorry, can you, when you're applying to whom for the approval?
Yeah, if you would like to apply a sort of a vector-based vaccine, which is, for instance, of animal origin, and you would like to apply it in human, is it easy to get the ethical approval?
Yes, I mean, I wouldn't say it's difficult these days because I think authorities are very receptive to new technologies, and I think there are now within veterinary medicine a number of vector vaccines using different platforms that have already been approved. And of course, once a vaccine's approved, it can be used freely in the marketplace. So if you use a vector that's already been licensed, then that'll obviously facilitate the particular process. But I think you just need to be careful of the choice of vector. You need to make sure that that vector doesn't spread or replicate within animals that may associate with your host. And so I think you need to be very careful in your risk assessment. And basically, when you go to register any new vector vaccine, you do have to do this sort of risk assessment to show in terms of this safety of that particular vector in its stability within the host, but also its ability to spread within a population. But it's not difficult. There are well documented and prescribed processes for doing it, and it's been successfully applied to a number of different recombinant vaccines now in veterinary medicine.
Well, thank you for that reply, Mike, and thank you to all of you for good discussions, and to the audience for getting us good questions into the room where we can discuss them. We will end the session of today. It's been a very nice day, and I have enjoyed it a lot. And this is, of course, thanks to the EuFMD team that has done a tremendous job solving all sorts of problems. And once again, I will mention that Pam Hollinger from California put the program together for this day, and thank you for that, Pam. The next session will be on the 17th, on Thursday, 17th of December. And you should be remembering that. That's session 4, Resilience to Long-Term Fast Crisis, the Importance of Animal Welfare, Supply Chain, and Business Continuity. So there is something to look forward to still, and that's nice. In the meantime, you can check out the presentations and the discussions. They will be streamed in the virtual room, where also all the posters will be visible, and you can go and explore these posters. Thank you, everyone, and over to you, Buddha.
Thanks very much, Sten.
Thank you.
Well, I think our presenters have left, but I was just about giving a very brief recap of day 3. I don't spend too much time, just in case if you have missed the morning session. I mean, the theme of this session today was vaccine security and critical resources for emergency management. And all our speakers gave very insightful views about different aspects of this theme, which is quite interesting. I will very briefly mention in the morning, we had 2 very interesting presentations on modeling and guidance of policy in relation to any investment in preparedness for emergency outbreaks and situations. Also, we looked at diagnostic supply chain lessons from COVID pandemic. That was very relevant talk by our keynote speaker. And then we had also we looked at also quality of vaccine, the immunogenicity and immune response of emergency vaccination for FMD in pigs. We also listened to our presenters from California, how California is being prepared for possible FMD outbreaks. And also, we also had a very interesting research being presented here on assessing the level of preparedness in European countries and how they are relied on EU vaccine bank. And in fact, that talk was very much relevant to our this afternoon presentation, starting with rapid vaccine development by Mike, and also a solution for vaccine security by Pascal, looking at vaccine banks and also the situation in endemic and versus free settings. And by Alistair, we listened to prioritization of vaccine and cost effectiveness of vaccination campaign. So very interesting, and that was very complementary to the talk by our colleague from Algeria on evaluation of human resources needed versus available resources. Human resources are quite often are neglected in terms of what is needed, and bringing private sector into play. And also we had a presentation on developing diagnostic test on ELISA in this case, which was very complementary again to all the efforts about preparedness and resource allocation, and also application of vaccine and findings of an example that was given by Dr. Recesor. So I think in a nutshell, we had very complementary talks on aspects of our vaccine security theme and allocation of resources, which I really hope the audience found it useful, and they can also look at this presentation again on our website. With those remarks, I would like to thank everyone for attending this session. And I give it the floor to you, Sten, for last words.
Well, thank you, Buddha, and thank you for your help as a moderator in this session. Thank you very much. And with these words, I'll close this session, and I hope to see you all again on Thursday, 17th of December. Bye.